Well, good morning. We'll go ahead and call our meeting to order. Great to see everybody again after the holidays. Hope everybody had a good holiday. So we'll start with the Pledge of Allegiance. If you would join me.
I pledge allegiance to the flag of the United States of America and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all.
Well, Josh, would you do the roll call, please?
Yes, sir. Good morning. Rachel Arizmendi. Ashley Bourne. Don Bransford. Don Cameron. Here. Nancy Cassidy. Helene Dillard. Mike Gallo. Crystal Haling. Eric Holst. Jeff Huckabee. Bryce Lumberg. Martha Montoya. Frank Mueller. Joy Sterling. Andy Thulin. Mr. President, we have a quorum.
Thank you, Josh. At this time, we'd like to have the departmental update. Undersecretary Jenny Lester Moffett. Karen's in D.C., correct?
Yeah. So thank you. Yeah, it's great to be here. Karen sends her regards. She is in D.C. today speaking at an event on the Foundation for Food and Agriculture, which is sounds just to be a really great event talking about biodiversity, healthy soils, a lot of the work that we're doing in ag and ecosystems work. Of course, the biggest update that we have at the department is that Karen has been reappointed. So she really sends her regards that she's not here today. One thing that we did do, and you guys all have a copy of it in front of you, is we pulled together the accomplishments that our team at the department has done over the past 8 years. I really encourage you to look it over. It's quite remarkable, the work that our staff has done, certainly at the helm, with the helm of Karen as CDFA secretary. So there's a lot in there on all of the different programs that we've been involved in, ways in which our programs have evolved over the past 8 years as agriculture continues to evolve and innovate as well. So it's a really great read, and I want to thank especially Jay Van Rain, who's probably not here, but with our public affairs team for putting this together. He always makes everything look really great and eye-catching as well. But all of our staff was involved in pulling this together, and of course, everyone at CDFA was involved in the work that came put into this report. On January 10th, the governor laid out his new budget, and in that budget, there were some key things for CDFA. Certainly, there was $565 million for the department that was proposed in the January budget. Some of the big things that were in that budget for CDFA is a new initiative called the California Biodiversity Initiative. This was laid out by Governor Brown in September of last year. Governor Newsom has taken it upon himself to put some funding behind that. So the department, he proposes to receive $4.2 million in general fund to really spearhead the biodiversity initiative, which would be an initiative that not only CDFA, but Department of Fish and Wildlife, as well as Resources Agency and the Strategic Growth Council have been partnered with. So it's very much an interagency initiative focused on biodiversity in the state, plant, animal species, developing things like germplasm banking and other things that are quite important, as well as a soils map for the state as well. That was a big one. There is funding again this year for the citrus pest and disease prevention resources, deferred maintenance. Safe and affordable drinking water was a big one, so there's funding for that. And then on just last Friday, on January 1st, Finance put out a trailer bill, a budget trailer bill language proposal for safe and affordable drinking water again. Quite a few members of the legislature have also put in drinking water bills. This is a priority of the governor. Actually, his first week in position, he, in the first cabinet meeting that the governor had on Friday of that first week, he brought cabinet down to the Monterey track and to meet with and to hear from people who have been without drinking water that that they're able to drink. So this is definitely a priority of his, and it's a priority that we're seeing through a lot of bills that are being proposed in the legislature as well. So we'll see how that all pans out. That's got certainly a lot of people interested in working on that as well, picking up where we left off last year, and maybe even certainly with the new legislature or new governor, there might be some new ideas floating around as well that we'll learn about. Healthy soils is also something that the governor brought up and mentioned in his budget speech, and he proposed $18 million toward healthy soils and referenced the Natural and Working Lands Implementation Plan in that as well. Then of course, there's continued funding from Prop 68 to continue to staff our state water efficiency and enhancement program, our deferred maintenance program for fairs, other things, of course, in our Healthy Soils program as well that receives Prop 68 money. Those are the primary things that were there. Certainly we have industrial hemp as a big priority. It's something that was adopted in the Farm Bill as a new ag commodity, so we're working through what that means for California. That will not only be in staffing, but also really looking at cross-agency and looking at our cannabis program and how those all work together. Legislation is being proposed daily. Rachel certainly has her work cut out for her. There is a lot of energy and excitement this year on legislation, and so Rachel is busy for sure. The last thing, and I hate to end on a down note, but in December, you guys have received updates from the Secretary on Newcastle disease. It's been going through backyard poultry in the Los Angeles, the Riverside, San Bernardino areas. Unfortunately, we had our first commercial detection in December. At this point, we have 5 commercial operations that have had detections, and so our staff and our state vet team have been working diligently with USDA, had several meetings back east with the USDA. Karen actually is today meeting with Secretary Perdue on this same issue, really making sure that we're working in tandem to really combat the disease, make sure we're containing it to the areas that we have it, and really working on making sure that this doesn't spread to other commercial and backyard operations as well, and other states for that matter. It has also transferred to, there's one case in Utah as well. So that is not to end on a sad note, but that is something that we are certainly working on and addressing at the department as well.
Are all 5 in the same local area, or are they?
Pretty much, they're all in the Southern California area. They're in San Bernardino and Riverside counties, primarily Riverside County. That's about 9, so between the 5 hen houses, that is about 900,000 birds. So very significant.
Thank you, Jenny. I appreciate the update. Yes.
I have a question. What is gene baking? Did you say gene
baking?
Did you say gene baking?
For the biodiversity, germplasm.
Thank you.
Banking?
Yeah, germplasm,
Yeah.
banking. That's a mouthful.
Thank you.
Thought we were cooking up something in the back here.
Yeah.
Don, question.
Yes, go ahead, Don.
I have a
Sorry.
question on the cannabis issue.
Yeah.
Is there enough funding for that, or is that going to take funds away from other programs? It seems that, and then I noticed now they're already talking about a tax break. So does that impact funding at CDFA? Or I guess my question is, are there adequate funding, appropriated dollars to manage that? And then if there's a tax cut, does that impact the CDFA budget in any way?
I don't know the answer for the tax break. I can get back to you on that. But yes, we do have adequate funding for staffing. It's not pulling from other programs. That money is coming from a whole separate account. It's actually coming from a loan from the primary cannabis account. So it's completely separate from all of our other ag activities. So, and we have built up a lot of staff there. They've been working hard. We had a December 31st deadline for getting temporary licenses approved, and they worked fast and furious all the way to the end working overtime all through the holidays. So certainly they received big kudos for all the work that they did, and they can have continued work to do now on annual licensing as well.
I'm sorry if I misunderstood. There is or there is not funding for Healthy Soils program?
In the governor's proposed budget, there is $18 million in Greenhouse Gas Reduction Fund proposed funding for the Healthy Soils program. It was between Prop 68 and Greenhouse Gas Reduction Fund, it was $15 million.
So,
An
3
increase.
million
Yeah,
additional,
an
yeah.
increase. And that 18 million, and the governor referenced this in his budget, is points to the Natural and Working Lands Implementation Plan. We ran some models through that plan last year on different acreage goals that we could get to, and one of the models showed that if we invested 18 million, we could get 500,000 acres by 2030 into healthy soils practices statewide just through the program, not including voluntary efforts, work through USDA, NRCS as well.
Great.
Yeah.
Yep, it's a good program.
Yeah.
Need to do a little housekeeping if we don't have any other questions. We need to approve the minutes from December 4th. Move to approve. Okay, second. Any discussion? If not, all those in favor signify by saying aye. Opposed? So moved. Okay. We're going to move on and get into the heart of the meeting here. Our meeting today is going to be discussing gene editing and more of a just a educational meeting for everyone to get everybody up to speed, bring the topic up, and have a little discussion. So our first presenter today is Becky McElprang from the CLEAR Project from UC Berkeley. Becky is a postdoc scholar from the CLEAR Project at UC Berkeley. The project, Communication Literacy Education for the for Agricultural Research, encourages young scientists to develop effective strategies for communicating science to a broad public audience. Becky's work has been included in the Conservation and the Berkeley Science Review. So if you would join us at the podium. Thank you.
Thank you so much for having me. So I'm a postdoctoral researcher now, working with Dr. Peggy Lameau at UC Berkeley. And in my time before that, I was a graduate student in plant biology at UC Berkeley. So when I talk about this stuff, it's from my experience as a graduate student, and then in the last year doing a lot of communication and learning more about the agricultural side. So before we get started, or before we go too deep into things, I want to talk about what gene editing is to get us onto the same page. Gene editing is changing the DNA in an organism at a precise location. So if you have hundreds of millions or billions of base pairs, with gene editing, we can now say, well, let's make a change at this one location. Maybe that's a location that's causing the plant to be more susceptible to disease. But we're able to use the CRISPR machinery, or the gene editing machinery, to make a change at a precise location. Now, there are three main techniques for gene editing. Talons and zinc finger nucleases were developed in the 90s and aughts, and they looked very promising for a while, but they have a couple of drawbacks that have made it difficult to scale them up. So when CRISPR-Cas9 was developed, or the gene editing capabilities of CRISPR-Cas were unlocked in 2012, it quickly became the leader of the gene editing techniques because of its ease and efficacy. So gene editing generally occurs in two steps. First, you have to make the cut at the precise location. And second, the change or the edit to the DNA must be made. So if we look at just the CRISPR-Cas system, we've actually been able to image what it looks like when the Cas9 protein is cutting DNA. I'm gonna try to hit the play button. Okay, so the red glob that I'm hovering the mouse over is the Cas9 protein, and the faint yellow strand is DNA. And if you watch it, you'll see that the Cas9 protein has cut apart the DNA at that location. And so now that the DNA has been cut apart, there needs to be an edit that's made. So what do we mean when we say editing? Editing can be the insertion or the deletion or mutation of just a few base pairs, or it can mean the insertion of a larger segment of DNA, perhaps a whole new gene. So I want to compare gene editing to genetic engineering and mutation breeding so that we can see the context within which different biotechnology strategies CRISPR fits. So like I said, with CRISPR, there's one type of editing where you make small mutations. Maybe you add a base pair or take a base pair out, or an A is turned to a T or something small like that. But with another type of CRISPR, another strategy, we can actually insert DNA into the genome at a precise location. So if we look at genetic engineering to get our bearings, or people also call genetic engineering genetic modification or creation of GMOs, we're also able to insert DNA into the genome, but we don't have the precision. We can't determine exactly where it goes. It goes in at a random location, and after the process, scientists can use sequencing to determine where that gene is sitting and if that's in a desirable or acceptable location or not. And I think that this conversation is well-rounded if we include mutation breeding. Mutation breeding has been done for over 50 years, where thousands of seeds are treated with a chemical, a DNA mutagen, or irradiated. And then the seeds are grown up in the field, and breeders walk through the field and identify plants with the best traits. That's how we got things like the ruby red grapefruit and thousands of other varieties that are grown today. One thing with mutation breeding is that it makes several mutations in a plant genome. And especially before the advent of the DNA sequencing technology we have now, we didn't know what all those mutations were, or even what any of them were. So who uses and who's regulating these different technologies? Well, genetic engineering is, of course, regulated by the Coordinated Framework on Biotechnology, as responsibilities are divided between the FDA, the EPA, and the USDA. And genetic engineering can only be used in conventional growing. Mutation breeding is used by conventional and organic growers, and it has no special regulations. And so what should the government do with CRISPR? And what is the government doing with CRISPR? Well, about a year ago, U.S. Secretary of Agriculture Sonny Perdue said that under its biotechnology regulations, USDA does not regulate or have any plans to regulate plants that could otherwise have been developed through traditional breeding techniques. So let's look back at this. Most people have assumed, and USDA decisions have supported, the idea that using CRISPR to make the small targeted mutations will not fall under the regulation of genetically engineered crops. There's a little bit more unsurety in the CRISPR and the DNA inserted at a precise location form of CRISPR. And the question there would be, is if the DNA that's being inserted is from a different variety of the same species, well then that could have been created with conventional breeding. So how will those be regulated? But most of what I'm going to talk about will be opportunities that we have to use this CRISPR for small targeted mutation to increase sustainability in agriculture. And the reason that I talk so much about regulation is because that really matters for how these technologies can be used. If regulatory costs are high, then developers who are working to create seed lines with these traits have to focus on crops with really high returns, like corn and soy. And that's what we've seen with genetically modified crops, that developers are going to spend about $130 million to take these seed lines through research, development, and regulation. And so they choose to use the technology on crops where there's a huge seed market, lots of farmers buying corn and soy. But if there's less stringent regulation, and when I say less stringent, I mean still appropriate and adequate regulation. Of course, it's the duty of the government to make sure nothing harmful gets into the food supply, but less stringent and appropriately so, less stringent regulation reduces that cost of commercialization. That means plant breeders who want to develop new traits have less developmental costs to recoup with their products. And so developers can use gene editing on crops with smaller markets and focus on regionally adapted varieties if the regulation is less stringent than with genetic modification. And again, I'm specifically speaking about the type of CRISPRed crops with the small targeted mutation, but this could apply to ones with gene insertions as well, or cisgenic varieties. And so if we decrease the cost, if academics and small seed companies and even big seed companies are allowed to create crops using this technology without the huge $130 million price tag, they will be incentivized, or at least not disincentivized, to create environmentally friendly crops. So I'm going to give you a few examples of what I'm talking about based on recent research. So the picture here I'm showing you is the surface of a leaf. You can see the different leaf cells, and I'll wave my mouse right here. These two cells, this one and this one, are called guard cells, and together they form the stomata, which is a small opening in the plant leaf. The stomata is important because it lets carbon dioxide in, which is then fixed and turned to sugars, and it lets water out. All of us recognize the incredible devastation that the drought like the one we suffered over the last years can cause. And so more stomata on a plant means that more water is lost. There's more little openings in the plant leaf for water to escape. Researchers found that by changing the amount that one gene, called PSBS, is how much that gene is activated, or how much we'd say it's expressed in the plant, really alters stomatal density. So they were able to increase the activity of the PSBS gene, and that improved water use efficiency. For each molecule of carbon dioxide that entered the plant, 25% less water was lost. Another set of researchers went a different direction, and they wanted to check out the same kind of idea, but they looked at a gene called epidermal patterning factor. They found that if they increased the activity or increased the expression of this EPF gene, they were able to reduce stomatal conductance, meaning they were able to decrease the amount of water that moved from the inside of the plant to the outside of the plant. Now, on the left in this graph, the EPF20E means EPF, or epidermal patterning factor, overexpressor. So that just is saying that the genes are overexpressed, or they're more active. The middle is showing the regular plant that's completely unaltered, and the bar on the right is showing when the EPF genes are knocked out of the plant, non-functional. So there is less water when we make the small change in epidermal patterning factor genes. They were also able to directly measure water use efficiency and found much higher water use efficiency in the plants where they had made this small genetic change. And very importantly, when they looked at photosynthesis, photosynthesis did not change, meaning that the reduced density of stomata and the reduced stomatal conductance did not translate to lower photosynthetic output or lower biomass for the plant. So another way that gene editing could be really helpful for sustainable agriculture is by increasing biodiversity. As we all know, crops have been bred for hundreds and thousands of years to generate the nice domesticated varieties that we grow and eat today. The things that we eat and grow today look very different from their wild relatives very often. And as we've gone through this selective process over the hundreds and thousands of years, we've lost a lot of genetic diversity as we've gone through this process. And in some cases, researchers have found that if you take the wild relative and you make a few small gene edits, you can re-domesticate the crop with just a few small gene edits, but you're bringing with it all of that fantastic genetic diversity that our modern crops lack. And I think Carl Gruber in this Nature article said it really nicely, so bear with me, I'm going to read this. Millions of years of adaptation to varied and often extreme environments has created a rich genetic diversity of wild relatives of modern staples. These plants represent an immense library of valuable traits with the potential to improve the quality and resilience of modern crops. Across the globe, researchers are trying to endow domesticated crops with these traits through interbreeding. Plant breeding programs benefit from such genetic diversity by creating new crop varieties that are nutritious, use natural resources more efficiently, and are able to respond to stringent environmental conditions and destructive pests and diseases, says Nora Castaneda Alvarez, a plant biologist. So to bring this kind of concept to life, on the left you can see a picture or a painting of watermelon from the 17th century. It doesn't look like anything that we find in the store. It's got a huge rind and lots of big seeds. And it may, but what we can't see are the traits that that watermelon has that weren't selected for consumer value, but that actually might have been very beneficial. Maybe it was especially adapted to grow in a region with salty soils or to grow and survive under disease pressure. But some of those traits were lost as we went from the picture on the left to today's watermelon. And so this isn't just theoretical, scientists are doing it. On the far left here is a picture of a wild tomato plant. And researchers at Cold Spring Harbor Laboratory mutated a gene called self-pruning in this tomato plant. And it went from indeterminate and kind of wild and all over the place to a much smaller, more compact plant that would be easier to grow in our agricultural systems. And moving over to the pictures of the flowers, the one with WT in the corner is the wild tomato variety. That's right here. And to the right, they've made one mutation, one small DNA change that has changed the wild tomato so that it creates many more flowers, and therefore would produce at a level that is more consistent with what we see in today's domesticated varieties. So gene editing can also be used for disease resistance. I'm going to use an example of cucumber and viruses. And part of why I use cucumber is because it's not the most agronomically important plant, but it does represent a good niche, and there is a significant market for it. And so it would be great if we could use tools to really enhance some crops that aren't, I don't want to say they're neglected, but they don't have as much focus as some of the more agronomically important crops. So viruses use host machinery for their own benefit. That's true in humans and in plants. And cucumber has a gene called ELF4E, which is used by cucumber viruses to help it make viral proteins. So if it's been a while since you've been in biology class, let's here's a little description of what I mean by that. So DNA in an organism is converted into RNA, and then that RNA is made into proteins. And proteins are what do the cellular work of our cells and make things happen. So if we can stop RNA from being translated into proteins, then we can stop the virus in its tracks. It doesn't get to reproduce, it doesn't get to go on to infect new cells. So this team used CRISPR-Cas9 to mutate the ELF4E protein, the ELF4E gene inside the plant. And that resulted in a stop of, that made it so that the virus could not use ELF4E to make its own proteins. So this table may look a little confusing, but let me walk you through it. Cucumbers with mutated ELF4E were resistant to 3 different kinds of viruses. The CVYV, or cucumber vein yellowing virus, was infected, and if it had a mutation in this gene, then after 14 days, there were no plants that showed any symptoms. Whereas on the far right, you can see that 60 out of 60 plants showed symptoms of the virus if they did not have this mutation. And similarly, for zucchini yellow mosaic virus and papaya ring spot virus W. This isn't a panacea. It didn't work for cucumber mosaic virus and one other. But what I love about the potential of gene editing is that we can respond quickly to problems where they're happening. And as a visual representation of the data I just showed, on the left is a non-resistant infected plant. In the middle is a resistant infected plant. And on the right is a plant that has not been infected. So you can see that middle leaf looks really good compared to its counterpart on the left. Those were a couple examples that I wanted to give you in depth, but there's so many other ways that gene editing could be used to increase sustainability in agriculture. So I've listed a few more of them. Things like increasing biodiversity by making orphan crops more conducive to wide-scale growing. Building soil health by helping plants recruit beneficial microbes to their roots. We can regionally adapt specialty crops, for instance, to grow in saltier soils. We can decrease pesticide use. Powdery mildew was what I studied in graduate school, and making a simple gene edit to reduce or eliminate the use of copper sulfates would be awesome. We can reduce food waste by making produce more durable for transport. For example, a simple gene edit makes tomatoes have the jointless quality. And when they have the joints, that means when they break off the vine, they have a little bit of a stem sticking up. And during transport, that can pierce other tomatoes and ruin the fruit. It can also help us adapt to climate change by making plant lines or plant varieties either more resistant to hot temperatures or by allowing us to transition growing regions. So maybe the plant can't stand the heat of the south, but maybe we can make some edits so that it's suited to the longer days and shorter growing season of northern latitudes. So I really believe that gene editing is a tool that can be used for the development of regionally adapted and sustainable crops. And I know that there is opposition among many people to using it, and I would just suggest that there's reasons to take a second look. With that, thank you so much, and especially thank you to my advisor and the sources of funding for the CLEAR project. So I'd love to answer questions now.
Thank you, Becky. We appreciate your presentation. We'll go ahead and open it up to questions from the board. Go ahead. We knew it was going to be
Thank you for a very clear presentation, very understandable. I have a question about your statement that the DNA is now able to be altered in a precise location. Do you have any evidence of off-target changes in DNA using the CRISPR technology?
Yeah, so there is, last summer there were a couple of studies that suggested that sometimes there may be more off-target mutations than were originally thought. I think that our sequencing technology is to a point, and especially now that we know that that's a possibility, that we have the technology, we have the capability to mitigate any negative consequences that could come as a result of that. I do think Richard's going to talk a little bit more about that.
Thank you for your presentation, Becky. Becky, I had a couple questions. One is, who decides if a genetically engineered plant through CRISPR is safe and worthwhile and releasable into the public or into the open? I have heard that the CRISPR technology is simple and easy and can be done in a high school lab or in a garage type of lab. And that would seem to me to indicate that you might be doing it in a controlled and direct kind of way, but who would keep somebody from, say, taking an almond in their backyard and taking it into a high school and doing a CRISPR work on it and planting it near commercial almonds and having an open pollinated plant do some crossing? Who decides where it can go and when it can go and keeping it out of an unintended situation?
That's a really good question, and something I think is really important to be thinking about, especially as I would guess most of us have heard around Thanksgiving time that in China 2 twin girls had their genomes edited, and that was against all of the international regulatory policies and what people had said was allowed. So I recognize the fear for that stuff, and I think that that's important. I think it's important that we have a regulatory system in place. I hope that for the CRISPR for mutation, it's more like how we regulate other crops that are more conventionally bred. A similar concern with the level of risk that there is with the CRISPR plants with small mutations, I would suggest that there's no more risk than somebody applying chemicals to a seed and going out and planting it and risking that being open pollinated, because we're just talking about small mutations that happen all the time through nature and that we've induced to have happen for decades. So while I don't want to say that there's no risk ever, I think that the risk of something like that is small, but it's small and manageable, I would say. And not significantly new or different with this technology.
Go ahead.
Thank you for the presentation. Obviously, the potential is endless in terms of this type of research, but I guess my question is more related to the control of these commodities that are generated, and not only from an identity preservation, and so seed pools don't get cross-contaminated or contaminated, because in California, a huge amount of our product goes overseas, and we have trade agreements, and USDA may make a ruling on this, but a trade agreement doesn't listen to USDA. So, what type of responsibility do the researchers have, and the seed companies that are going to generate this in terms of protecting, the grower from some sort of trade embargo? It happened in rice. There was GMO rice developed. It was contaminated, and overnight a market shut down. The EU shut down the rice market.
From China,
So,
is that
so what kind of controls do you all have even internally as a researcher? And then beyond that, what kind of controls the seed companies have to protect all of us?
Well, I think that's a good question. One of the things that makes it difficult is that with the GM rice that was imported, mixed with conventional that was imported into the EU, there was a test that they could do. There's the transgenic DNA or the foreign DNA that can be tested and assayed for. With gene editing, there really isn't a way to test for it. And because it's such a natural thing, it could have happened just in nature. It's no different than the plant making an error in DNA replication, as plants do, as our cells do, as everything does when it creates the next generation. And so there may not be a precise way to detect these changes when they're made from small mutations, because they look 100% like a natural counterpart. I think that that points to the risk of these. They're indistinguishable from their natural counterpart genetically. And so what risk are we taking on? And I would argue that it's quite small. It looks 100%. It looks just like the naturally bred variety. And the single mutation that it has could have happened in nature. So I know that as researchers, we are very cognizant and aware and have very strict regulations on ourselves and our work to make sure that we're not spreading anything that hasn't gone through appropriate approvals. The companies will have to speak for themselves. I don't know their policies on how they do that. And in the end, I think the farmer gets to decide what to grow. And the farmer will have to evaluate that risk. I mean, policy and government, I hope, will do what they can, but trade law or trade deals is not my area of expertise, so I don't want to go too far into that. But I think that it doesn't hurt to give farmers a variety, an option that could be better for them and their farms.
The one issue is, you know, a country is going to say, you have to prove there's no CRISPR seed in this, or you can't export it. There are phytosanitary conditions that get set, or protocols that get set up. So if there's no test, how do I as a grower prove there's no CRISPR seed in that?
I think you have to trust the breeder that you're working with and the people that you're buying the seeds from.
If a trading company says you have to prove it, and I can't prove it, then gone, market's gone.
So I guess then the farmer would have some responsibility when they purchase seeds to say, I want to know how this was bred. Because we're not going to try, I don't think that in the United States we're going to try to hide that, or that anybody's going to try to hide that. So I think from our side, we have good opportunities. We have companies that we can regulate and produce the documentation of how individual varieties were bred, and those could be passed on to the farmer. But that is a little bit outside of my wheelhouse.
Hi, thank you for a very excellent report. A question for you: when you talk about the regulation, is the approach of not regulating one that has been adopted in other areas? How has the EU, for example, chosen to regulate?
So the EU came out and said last year that they would treat these types of plants with the small CRISPR mutations the same way that they treat genetically modified foods. So that is something that needs to be considered with growers who grow crops that are exported. That is what the EU has said. A lot of countries have not made statements on it yet. Canada is treating it the way that it looks like the US is treating it, and I believe so is Brazil. So there's a difference in how regulatory agencies across the world look at things? Do they look at the product or do they look at the process? And if they're looking at the product, then they view this like any naturally bred type of plant because it is genetically indistinguishable from a naturally, conventionally bred plant. But countries that are really concerned with the process, like the EU, or not the Union of Nations that make up the EU, have decided that they care more about the process. And so they are choosing, for now, to look at these as genetically modified. I will mention that the chief panel of scientific advisors to the EU came out with a statement saying that that policy did not have a scientific basis.
Good morning. Thank you for your presentation. So I have a question. My question is, with the GMO, traditional GMO, a lot of it's been used to build chemical resistance into different varieties of glyphosate resistant things. Can the same thing be through CRISPR? Can that also be done? Because we've seen resistance to these pesticides build up in plants, so you could argue that the CRISPR technology would allow that to happen also?
CRISPR technology could allow that to happen, just like conventional breeding could allow that to happen. There are plant varieties out there that have been traditionally bred for resistance to such things. So certainly that's something that could happen, but that's not unique to CRISPR.
Yeah, I was interested in the tomato case that you mentioned, taking the wild relative and modifying it. So I guess the answer to this might be the same, but presumably there's some risk then of that new genetics entering back into the wild if there aren't precautions taken. So has thought been given to, first of all, is there a way to sort of turn off the reproductive organs so that you don't have pollen getting back into the wild, or presumably there are policies that could be instituted about where those modified varieties are planted so that they're not mixing with wild. But I'm just wondering what thought has been given to that.
Yeah, well, the tomato variety is a good one. The mutations that they made were actually the same mutations that already exist in conventionally bred varieties. So in the gene-edited plant, you have some genes that are from the wild tomato and some, a couple mutations that are found in the varieties that we've bred over hundreds or thousands of years. And so there's really no greater risk of mixing with the wild population because everything in that tomato is either from the wild population or already exists in our domesticated varieties. And so it's had a chance over hundreds of years to do that crossing out into the wild varieties.
Can I just ask a follow-up on that? So what was the starting plant in that particular case? Did you start with the wild?
Yep, they started with a wild plant. I can't remember the exact scientific name for it, but I can certainly give that to you. But it was a wild tomato variety.
And then they inserted a handful of genes that...
They didn't insert any, they just made a couple mutations in genes. So there's one gene in the wild tomato that makes it have kind of wild, out of control branches. And so they just made a small mutation in that gene so that it didn't work very well anymore. And that actually happened naturally a couple hundred years ago as we domesticated tomatoes. So the benefit is that we get that mutation, but we also get some of the disease resistance and other traits that that wild variety has to offer.
If you're comparing it to conventional breeding, I'm wondering if you're starting from pure wild stock and you're making edits, is it fair to say that resulting edited genome is closer to the wild relative than a conventionally bred tomato, which might have been altered over 100, 150 years? So the point I'm making is that because of the precision of this technology and the rapid turnaround on new genetics, the risk might be higher that you would sort of taint wild populations with basically human-created genetics, which, you know, I mean, we could talk about whether that's ethical or not, but it just seems a little bit closer to sort of monkeying with wild biodiversity as opposed to conventional breeding, which I guess its advantage is just slow.
Okay, so let me make sure I'm understanding you. You're thinking that because the wild crop with gene edits is more similar to the wild variety, it would be more likely to outcross with the wild relative and take on some of those domestic traits. Sure, that is a thing that could happen. That's also a thing that could happen with other breeding, well, okay, two things. It could also happen just by chance in that wild plant because it is a small mutation, and it certainly has happened in the past in the wild relatives because there's so many out there and mutations happen quite regularly. The other thing is that this strategy is being used by conventional breeders as well, where they're taking the domesticated variety and back crossing it to the wild relative in order to get some of that genetic diversity from the wild relative. And that can have good results, but it's also a really random mishmash of genes that you get when you just do that cross, and takes a lot more generations to work out what good traits you want into your variety that you're developing, as opposed to just making a few edits. So sure, that's a possibility. I think that it's not going to cause a problem that hasn't already happened.
So this is a clarification question for me. Am I correct in hearing that you are making a distinction between small edits, mutations, and inserting different genes?
So the distinction I'm making is between inserting genes and then making a small mutation. A small mutation is a small edit. It's the same thing where you're inserting or deleting a couple of base pairs or changing one base pair to another.
So it sounds like you've been saying that the small mutations are not a concern because they're very similar to what one could do in conventional breeding. Are you also saying that the idea that there's no or low need for regulation or attention to insertion of DNA, and is there a way to measure or test for that?
So what my opinion would be on this, and this is just my opinion, that based on what the USDA said, that they will regulate plants, that they're looking at whether the plant could have been created through conventional breeding or not. And if it could have been created with conventional breeding, then they're going to not give it the GMO regulation. And so for that insertion question, where we're inserting a gene, it really depends on what the nature of that gene is, in my opinion. If that is a wild tomato gene for disease resistance, that we could have taken the domestic variety and crossed it to the wild variety with the disease resistance, and then selected for disease resistance over, half a dozen generations. That is something that could have been developed through conventional varieties. So my interpretation of the Secretary's statement would be that that is not regulated like a GMO, but I'm not certain that that's how it will shake out. Anything that is inserted that's from a different species would certainly be regulated like a GMO.
I think my question was a lot on the same line as Crystal's. You'd mentioned there's 2 types of CRISPR, or maybe there's 10 types, but you were breaking it out, I think, into 2. Are you advocating that the more simplistic CRISPR is an appropriate technology and that more complex are maybe not, and that the older GMO is not? I think in the early discussions of GMO, the proponents were saying this is the most precise, and we know exactly what we're doing, and it's better than traditional breeding because we know what we're doing and why we're doing it. And this sounds to me like now CRISPR is better and newer, and that you're advocating for it and not for the stage 2 CRISPR or the GMO technology. And I was wondering if you could speak to that must be a wide range of CRISPR, and your presentation was really just about the most simplistic.
Yeah, thank you for asking that question. I do support genetic modification as it has been done. I do support the type of CRISPR where a gene is inserted from a different species, as long as the precautions and the testing are done to make sure that it's safe for humans and the environment. And I do have confidence in our regulatory system as it stands now that it does do that job. So I'm being an advocate for one type of technology and saying we can use it for sustainability, but I'm not trying to throw the other one under the bus. And I'm sorry, you'll have to remind me the second part of your question.
Well, I think you covered the answer.
Okay.
Do you foresee a CRISPR camp and a GMO camp in this process, since it's a little more clear-cut as to what's going on, and it falls within the USDA guidelines where GMO does not? Do you foresee a bifurcation of what's going on, and who? But that's enough.
Yeah, it's hard to predict that or foresee what might happen. My hope is that some people who are wary of genetic modification might come to see this type of CRISPR for small mutations as a really promising technology that they don't want to miss out on, because it could help them reach the goals that they're seeking in terms of sustainability and taking care of the soil and all of those things. I think that these traits could really be helpful for that. So that is my hope. I'm not counting on that. The National Organic Standard Board has rejected gene editing as a tool that organic growers can use, at least for now. So I see that people who support people who accept the safety of genetically modified foods and crops certainly tend to embrace CRISPR. What I'm wondering and what I'm hoping is that people who are a little bit skeptical or wary of GMOs might see CRISPR as a really great technology that could help achieve their goals.
So Becky, thanks again for a great presentation. So in terms of the work that the CLEAR project has done with a variety of groups, what has been kind of the response in regards to this issue from the groups? What's the public sentiment, so to speak?
The public sentiment is varied and often strongly felt. A lot of people are really open and curious for more information. And it's hard to embrace or accept new technologies when you don't really know what's going on. And I think we can't ask people to embrace or accept new technologies when they have no idea what's going on. The picture of a syringe on a tomato sticks out in my mind is something that's often shown, and that's not what's happening. And so what I love about the CLEAR project is that we're really trying to close that gap, and we're not trying to tell people exactly how they should think. I'm here and I'm stating my opinion and advocating for what I believe. But when we talk to the public, and especially when we talk to young adults or high school kids, we're not trying to tell them what to think. We're trying to give them all the information and tell them that they should think about it and that they should be part of the conversation.
I just want to challenge that a bit, because you did make the statement this was precise technology, and then you admitted it isn't. So I think we have to be careful that we don't represent something as...
I'm sorry, I don't know what I said to suggest it was not precise.
I asked you if there were any off-target changes, and you said yes.
Oh, So in some cases, there may be off targets. That seems to be a big exception to the rule. And, you know, I think Richard's going to talk more about that. That's something that he does every day, so I'll let him speak more to that.
Becky, I had a real quick question, we'll wrap it up, but, you know, it seems like the GMOs that came along were all grower oriented. Do you see that, you know, some of the traits with CRISPR are going to be more consumer targeted in the future?
That's my hope, and that's what I'm starting to see, and I hope that that does turn out to be the case.
Well, I thank you for your presentation and answering all our questions and bringing us up to speed on CRISPR. So thank you. Our next presentation will be Dr. Richard Michael Moore from the Genome Center, UC Davis. Dr. Richard Michael Moore is a professor in the Department of Plant Science, Molecular and Cellular Biology, and the Medical Microbiology and Immunology at UC Davis. Dr. Michael Moore is also a director at the university's Genome Center. So, welcome.
Great. Well, thank you very much for inviting me and an opportunity to speak to you. And also thank you, Becky, for a great introduction. We did share slides beforehand, so we're coordinating a little bit. And some of your questions were great. I hope I can address them as we go along. Also, I'm going to cover quite a lot of ground here. I've got 23 slides, I think, but I'm very happy if you wanted to stop me and we can discuss an individual slide as we go along. It's sometimes difficult to sort of jump around. So we're going to cover a lot of ground. And as I say, if you want me to stop and we can discuss individual slides, I'm very happy to be interrupted. So we're at a very interesting time in biology in our ability to characterize genetic variation and then to manipulate it. What I want to do here is to put CRISPR in the context of modern plant breeding and potential and look forward to where I think we need to go. So, you know, what are the current challenges facing us as a society, how to improve our crops? How can, you know, what role can plant breeding play in that? And particularly, what are the new opportunities presented by a number of different technologies, particularly genome editing? So, you asked me somewhat to talk also about global warming and, you know, the role here. I hope we don't have any disagreement the fact that global warming is a reality. Contrary to some wishful thinkers, possibly, global warming is a reality, and we're going to have to change this. We're going to have to address this. This is going to have a major impact on crop productivity. There are numbers of diagrams you can see. Here we have one showing if you live in Canada or Norway, it could be a good thing. If you live in the rest of the world, it's not going to be great, okay? And agriculture, global food security, food supplies are absolutely critical to political stability. We're not going to solve the political consequences of immigration unless you make it so that people don't want to move from where they are. And so the opportunities to increase food supply, we're going to have to increase food supply significantly. And I think plant breeding has the opportunity to contribute to that. Getting closer to home, there are a number of studies of the impact of global warming on California. This was a study done by a group at UC Davis. You can see different regions of the Central Valley, the impact of different crops, and you can see some crops basically are going to be wiped out in that particular area in California. So it isn't just a matter that we want to increase food security in less developed countries, sub-Saharan Africa and places like that. It's going to have impact locally as well. So what are going to be the impacts of climate change on California agriculture? We're going to have higher summer temperatures, and that means we get poor flowering and fruit set. the particular part of the plant life cycle that is sensitive to high temperature is flowering and fruit set. So we're going to have problems there. Also, you're going to get fewer chill hours. A lot of fruit crops need hundreds of hours below a certain temperature, otherwise you get very poor fruit set. We've already seen that a little bit. The pistachio, was it in 2016, had low chill and had a very poor crop as a consequence. This is going to be a recurring problem. We'll get extreme weather fluctuations, unstable food supply. We'll get reduced snowpack. Difficult to believe this week, but it's going to happen. You see various models where you get very little snow up there, which is going to mean less water for agriculture and the urban use. We'll also get greater losses due to viruses and insects. As things get warmer, you get insects moving up from the tropics carrying viral diseases that we haven't had before. So to get a sustained food supply, we're going to have higher, more stable yields grown on less acreage with less water, less fertilizer inputs, and hopefully minimal ecological impact. These are all challenges which we need to, this is reality, this is what we need to address. I believe it may be possible, but we've got to use all the tools that we have available. This includes GMOs, it includes genome editing. If we want to take a responsible attitude to the future, we've got to use everything we have available. Genetically improved crops, therefore, are the cleanest and most predictable element, but they're only part of the picture. We're going to have to make improvements all the way out throughout the food chain, both pre-harvest and post-harvest. There's a lot of potential, there's a lot of food waste that we can address some of the shortfall in food if we address the post-harvest issues. So I want to be quite clear, genetic improvement is only part of the solution, but I think it's an important part. Traditional plant breeding you can think of as a cycle of increasing and decreasing variation. And I've shown on this diagram, the green circle, that's traditional plant breeding. Basically, you identify useful variation, often in wild species. You make a cross, and that introduces a tremendous amount of genetic variation, most of which you don't want. And you then may make another series of crosses, but you then make selections from the variation you've got that hopefully is an improvement of what you had before. And then you do variety trials, you get improved variety, that will then be released. But then you go around, you're continually, you cannot sit back and say, we've achieved what we need. Plant breeding is a continual process, so you get this cycle. Now with various types of biotechnology, you can increase the variation, but you're still going around this cycle. And putting novel transgenes with GMOs is just part of that cycle. So, next generation breeding. We have a whole series of challenges and uncertainties. That's why it's a continual process. We've got climate change that we just mentioned. We have variability in existing pathogens. Crops are continually challenged by pathogens. And just like the human flu virus, they change, right? So you're always trying to keep up with them. We get new exotic pathogens. You get them spread around the world. You see them jump into new areas and cause new epidemics. We've talked about reduced water. We have nitrogen application issues. We don't want to contaminate the water, the groundwater, so we need to have crops that are using the nitrogen applied much more efficiently. We have changing consumer preferences. Breeders have always got to watch out for people suddenly decide they like bagged salads rather than crisp-head lettuce. Probably a good move, given the crisp-head lettuce. I'm a lettuce geneticist, so, you know, I can. Labor costs, you know, what's the opportunity for mechanization? Food safety concerns. We've obviously had a number of outbreaks with E. coli and Salmonella. You know, these are things that we can factor into the breeding goals. We have regulatory constraints, GMOs. I mean, don't get me wrong, GMOs should be regulated. but they need to be part of the picture, in my opinion. We've got industry consolidation. There used to be large numbers of breeding companies. Now there are fewer and fewer. So this is another thing that sort of impacts what breeding happens. We also have limited funding and resources put behind breeding. At the moment, to go around that cycle takes a long time. We need to speed that up. And also germplasm access through the CBD and the Nagoya Protocol, which I'll discuss in my last slide. However, we do have a number of opportunities. We have increased access to genetic variation with the new technologies. We have increased knowledge of the important traits. Molecular biology is really being able to go in and tell us what the critical genes are in a way that we really didn't have. And this is based on fundamental research on model species, often not the crop species. We also have increased computational power. We can do things, we can model things that we never had the ability to do even 5 or 10 years ago. We also should look towards precision agriculture, that smart agriculture where it's much more efficient using the sort of data-driven, knowledge-driven agriculture. And then there's vertical farming, indoor farming. These are all things that are on the horizon. And as a plant breeder, given that it takes 5, 10, 15 years to put something out, we need to be sort of looking into the future as to what we can do. And then there are a series of new technologies, which I'm showing here. So there are a number of contemporary transformative technologies. High throughput phenotyping, that is drones. You know, you see people love drones, but they can be very powerful and very informative in agriculture. You can fly and get phenotypic information on a scale and an accuracy that we never could before. We have molecular markers in that we can characterize genetic variation on a granularity that was inconceivable 5, 10 years ago. We have high throughput DNA sequencing. We can really characterize what is in the seed in a way that we could never do before. And this discussion we had about off-target effects and things like this, we can actually go in and ask, are there genetic changes there that we didn't know about? We couldn't do that before. And there's an appreciation that even within a cultivar that we think is very uniform, you'll find there's a fair amount of genetic variation there. We can take annotated genome sequences. We can now sequence very quickly any crop species. And it used to be we would sequence a species. Now we're sequencing hundreds of cultivars of every species. All of this results in unprecedented amounts of big data, and our ability to handle it is a challenge. So I've been talking about that top half is genetic variation. That's kind of the input. What's really interesting now is we have the unprecedented ability to manipulate, to actually use that genetic variation. Through inexpensive high throughput genotyping, we can characterize populations in tremendous detail. And we then use that for marker-assisted selection. That is, we can make informed decisions on the genetic variation. There's genome editing, which I'm going to spend quite a lot of time in the coming slides. And also there's microbiome exploitation. People forget that much of life is not what we see. You may have heard the statistic. I mean, if you look around the room, you don't realize, but actually you're just bags of bacteria. There's 100 times more bacteria within your skin than there are human cells. And plants are just the same. They live in a very complicated environment, and actually roots are much more complex than the human gut in terms of the microbiome. And there's a lot of opportunity for manipulating that. And actually, in Northern California, there's quite a few small startup companies looking at the microbiome and the potential for manipulating agriculture to agricultural benefit. So these are all things we have to think about, but I'm going to focus on genome editing as I was requested to do. So going back to this slide, what's really interesting about genome editing then is because of its precision, you're not really getting explosion of variation when you make a cross. You're going in and you're making a very precise edit. There may be a little bit of background variation, but it's nothing compared with the type of variation you get when you make a conventional cross. And so what you're doing here is you're really interrupting, it's eliminating the breeding cycle. You can really go in and make very precise changes that then will allow you to go to field trials. And yes, you can sequence it and you can see whether any unintended genetic changes. And you could make a few crosses just to clean that up. But this is really going to speed up our response time to change. So, genome editing. This is really a transformative technology that's revolutionizing medicine and agriculture. And whether you like it or not, it's going to happen. The question is, how do we harness it to society's benefit? The thing about genome editing, the CRISPR system, it makes a precise cut in the genome. And that's all the enzymes are doing. They are cutting the DNA. It's then the edits is really harnessing natural processes that are occurring in the cell. So some people say, well, what does CRISPR stand for? You can tell I'm not from Kansas. Crisps in England are what you call potato chips. Thank you. Yeah, crisps. So anyway, CRISPR actually stands on the left-hand side of this slide for clustered regularly interspaced short palindromic repeats. Right? I have to have this slide. I can never remember. But what they've done is part of the defense system of bacteria, in that bacteria, as they say, big fleas have little fleas on their backs to bite them, little fleas have little fleas, and so on ad infinitum, right? So that occurs in biology. Bacteria may be pathogens, but they have pathogens. And the bacteria have viruses. And the way bacteria defend themselves against viruses is that when they're invaded, they can take a little piece of that virus and they put it into their genome. And then that's used as a sort of, as a mugshot of previous invaders. So it's a very sophisticated system. And then the bacteria patrols its inside the bacterium. It patrols and said, am I being attacked by anything I've seen before? Okay? And if it is, it cuts the piece of foreign DNA. So what has happened then is people appreciate that this is incredibly precise. And so they've harnessed this bacterial defense system to engineer any piece of DNA. So that's why it's called CRISPR. The other thing here is there are many, many defense systems. Bacteria are incredibly diverse, and you will hear of CRISPR-Cas9 was the first one, but there are many, there are literally hundreds of systems that have been developed, that have been evolved over time in bacteria, and different companies are exploiting these different enzymes. So, CRISPR-Cas9 was the first one that came along. It was developed by Jennifer Doudna down in Berkeley. As I say, all it does is it cuts the DNA. So the protein is just a generic DNA cutter. Its specificity is driven by the fact that it globs on to a piece of RNA, that then another piece of RNA, which is complementary to your DNA, that is what gives it its specificity. And this is the power of the genetic engineering, is that this protein here is generic, and this specificity is just through this short piece of RNA. And that is easy to make. So in terms of the ease of engineering, you don't have to change anything. Proteins are difficult to work with. All you have to do is to synthesize chemically a short piece of RNA. And then this will attach to the protein, it will cut, and it will cut at this site. Now, the off-target effects you're talking about is when occasionally this will go down, and if you haven't selected your site accurately enough, it can cut somewhere else. So this is kind of the off-target effect. Having cut then, that's all it does. There are two somewhat different natural pathways that will repair. And I don't want to go too much in the detail, but One is if you cut the DNA as shown here, if there's no other donor DNA around, the natural process is try to join the chromosome back up together again. And when it does so, it can do that somewhat. It's not a perfect process. If it cuts the object biologically, as far as the cell is concerned, it's trying to make the accurate cut again. If it does that, the enzyme cuts it again. But occasionally, there'll be a bit of a wobble in the repair process. And so it'll either put a different base in, or it'll make a deletion or insertion. And when that happens, it'll no longer cut, and you've got your edit. Sure.
I just have a quick clarifying question. So does the process presume that you've already sequenced the genome of whatever plant you're?
Exactly, yeah. So much of this is enabled by the DNA sequencing technology. You wouldn't be able to design your guide RNA unless you know. And it's more than just having the sequence. It's having knowledge of what genes you want to
And
act.
what the sequence of that gene.
The
So
sequence
you
of
can
that
design
gene.
the snipper based on a
Exactly.
precise- Okay.
Exactly. Good question. So in the other way, though, that Becky was talking about, the second type is if you want to make an insertion or actually change the sequence specifically. If you have a template, the cell will look around and say, I want to repair this break, and it'll take another piece of DNA and repair based on that DNA sequence. And so you get sort of this is the more common one, and this is sort of the prevalent type. What we really like to do is to increase the efficiency of this pathway, where you can add a piece of donor DNA. Let's say you've sequenced a beneficial gene from a wild species. You look at that sequence and say, I want that sequence in my cultivar. You can then add that in, and it will convert the sequence in the cultivar to the new sequence. This is relatively inefficient at the moment. But the technology is going to be there in 3 to 5 years to do this. Or also, if I want to, I can actually insert a completely new piece of DNA in there. So I can say, I want this piece of DNA from a wild species. I add in either side sequence similarity here and here, and I can drop that sequence in. But it's pretty inefficient at the moment.
And do you need any reference to the wild species, or can you design that DNA sequence in the lab? In other words, do you have to go into the wild, grab the gene, pull it from there, and physically?
No, you don't have to actually grab it. You synthesize it in the lab. But I could synthesize any sequence I like, right, and I could stick it in there. The chance of it being biologically useful is essentially zero, right? So what you need is the knowledge from the wild species saying this is a good gene. I want this sequence, right? Yes, it's knowledge driven. But you don't actually physically have to necessarily. Sometimes it's easier if it's a big piece of DNA. It's easier to go in and amplify it up from the wild species rather than synthesizing it. So, the way I like to think about genome editing, I think there's 6 aspects. One of which is having genes to edit. Right? Getting back to your question, we need to know what our target should be. And there are some genes that are agriculturally useful if you knock them out. But that's relatively small number. Biology doesn't work. Things have come along to be useful. And therefore, in many cases, we actually need to have active versions of the genes rather than just knockouts. We need efficient genome editing technology. I've just explained knockouts are easy, but they're less agriculturally useful. So what we need are methods for gene replacements and insertions. And that's coming along. My lab, but many other labs around the world are working on this. We also need non-DNA ribonucleoprotein strategies. So at the moment, the way that we make edits is we actually introduce a piece of DNA into the cell that then the protein's made, the RNA's made. It'd be more efficient if we could actually in the test tube, make the protein, add the RNA, and there's no DNA involved. And there's no opportunity then for DNA being, extraneous DNA being integrated into the chromosome. I would like also methods for introducing the editing reagents, not going through tissue culture. At the moment, you often have to go through tissue culture, and that's slow, and again, you can get some inadvertent genetic changes due to tissue culture, not due to the editing process. And also, it would slow the speed down. And there are a number of technologies that are really exciting out there where we can actually change the plant as opposed to, again, it's all a matter about response time. How can we be flexible in responding to the myriad of challenges we're faced with? Then we have intellectual property issues. We could spend all afternoon on that. It's rapidly evolving. You definitely got to have freedom to operate, and a number of companies have powerful positions, shall we say. But interestingly enough, because there are more and more editing systems out there, it isn't that there's only one technology and somebody has a complete lock on it, which is healthy. So there are a number of ways of getting at editing events, and the more there are, the more, the cheaper the barrier to getting a freedom to operate goes down. And then there's regulatory issues, which we've discussed, and I'll be happy to discuss more, whether it's a GMO or whether it's not GMO. And then there are ethical and safety issues as well. I think all six are very important. So we were talking about production traits versus consumer traits. I've listed here some production traits. You'll be good to hear there's some consumer traits on the next slide. I couldn't get them all on one slide. So these are not things, there's a couple of editing traits that are close to market, but these are things that are definitely feasible with editing. You can manipulate flowering time. The examples of this in the soybean and tomato put in brackets the gene that gets edited. So, the molecular biology was worked out in model species. We can say if we knock this gene out or if we manipulate it, the chances are it will have a benefit in the crop. And therefore, these changes in phenotype have been demonstrated in all of these crops. And there's some very interesting ones. You can change the architecture of maize so that it receives the light more efficiently. Brassica oleracea, one of the big problems in many crops is the seed will shatter and you lose that. But that's controlled by a single gene. If you knock that out, the seed doesn't shatter. Dormancy in barley. We've heard some examples of disease resistance already. Bacterial blight, mildew in wheat, bacterial canker in citrus. We heard about herbicide resistance. We're talking about that. There's a gene called ALS, acetylanthosynthetase, that's involved in amino acid production. There is naturally a single change to the protein that occurs naturally, occurs in weeds already. And there are some conventional crops that have been moved to be herbicide resistance through natural breeding. Editing, we could make those events quite easily if you wanted to. There's drought resistance in maize. But I agree with you thoroughly, it's extremely important that we have consumer traits. And this is just a partial list. I've given you a reference at the bottom for more. But phytate content in corn, phytate complexes, phosphorus and availability, you can change the oil content in soybean and other oil crops. There's low amylose rice, just knocking out, you can change how glutinous and the starch content of rice. There's modified starch in potatoes for industrial starch. Cadmium in rice, cadmium accumulation is not a good thing. Ascorbate levels in lettuce, they're all imminently manipulatable. These are not things that are near to market, but it's just an example of things that could be done. And what's interesting about CRISPR, it really was only just revealed in 2012, and the adoption of this technology is really impressive, that the potential of what's taking off. Here's just a little example from my own lab. Lettuce will not germinate. Conventional lettuce does not germinate above about 25 degrees C. But that's a problem down the Imperial Valley and in Yuma. When they go in, thermodormancy allows for very poor stand. What's interesting, a colleague of mine, Louise Jackson, noticed, actually, if you are driving around here and you see things that are about this high, they're bright green, looking pretty healthy in August, that is actually wild lettuce, completely sexually compatible with cultivated lettuce. It doesn't mind the high temperature. And so a colleague of mine took some seed from an abandoned gas station in Davis, and when we germinate that seed, it's fine at 35 degrees. And collaborating with Ken Bradford, we found that it was a single gene that had been knocked out in the local wild lettuce. And so what we did then, we said, okay, we know the phenotype. Therefore, to proof of concept, we knocked that same gene out in a cultivar. And you can see on the left, it doesn't germinate at 35 degrees. On the right, we've just knocked out a single gene. It now germinates perfectly well at 35 degrees. So it shows you, if you have the fundamental knowledge, that you can radically change the performance quite usefully of the crop. So, as I say, we're at an interesting juncture because we now understand increasingly the numbers of genes that we'd like to put into the crop or change. And you say, well, I'd like a little bit of this chromosome, I'd like a little bit of that chromosome, I want a little bit of that chromosome, I want a little bit of that chromosome. Rapidly, it becomes impossible to combine all the good genes using conventional breeding. You'd have too many crosses, too big a population. And then even if you did get 8 or 9 genes into the same cultivar, as soon as you make a cross, everything falls apart again. Right? So what do we do? What we have to do, I believe, and this is going to be a transition for the whole of plant breeding and cultivar production, is we need to make gene stacks. That is, we need to take all the genes that we like and put them in a single position in the chromosome. And then it gets inherited as a single, what we call a single Mendelian unit. And in fact, probably we will put in a herbicide resistance gene into the middle of that, even if it's not used for commercial production, but it will allow the breeder just to spray with herbicide, and they've got all the good genes. And then they can get on and do the more difficult plant breeding. So if you can fine-tune then the crop fairly rapidly for a particular area, for a particular challenge, if there's a new disease threat, it'll allow much faster response time. We'll need efficient editing, I've talked about. It needs to be genotype independent. And I say we need to avoid tissue culture. So particularly with disease resistance, it's never a static process. I mean, just like the human flu virus, it changes. And you put a selection pressure on any pathogen, it will change. And if you think of the number of plants versus the number of pathogens, who are you going to bet on? You're going to bet on the pathogen, right, every time. However, what we want to do, so you have this cycle, so-called boom and bust cycle. It's been observed in wheat production since the 50s and 60s. You have a susceptible cultivar, you screen your germplasm, you identify resistance, you cross it in, you breed it, it's successful, the acreage of that cultivar goes up, that puts a selection pressure on the pathogen, it becomes susceptible, right? The pathogen changes, boom, it crashes. So you get this boom and bust cycle. This is sort of characterizing breeding for many plant diseases. And the moment, the equilibrium is pretty much to the left, right? It favors the pathogen. What we need to do is to push that to the right, okay? We want to sort of push that to the right. We'll never get rid of it, but what we want to do is to favor pushing it to the right. And one of the things we want to do then is to make the evolutionary hurdle for the pathogen as big as possible. And one of the ways of doing that is to have many resistance genes. If we release one resistance gene at a time, the pathogen will evolve quickly. And sometimes it only takes 2 or 3 years, even before the cultivars really got popular. But if you have 4 or 5 genes, then the evolutionary hurdle is way bigger. And hopefully, it'll be fairly successful. So what we're trying to do again is to increase the stability of the food supply. And this is, so we want to make cassettes of resistance genes. So where my lab and several other labs are really driving towards is that we'll be able to clone resistance genes, and then using genome editing, I'm going to put them in the same position beside each other on the chromosome. I'll have a herbicide resistance gene in the middle, and then when I release this, so I don't release cultivars, I release advanced breeding lines, and then the seed companies take that up. And then if I was to release a cultivar with 3 resistance genes on different chromosomes, as soon as the breeding company makes a single cross, my whole pyramid has fallen apart. But here, I put them side by side. They cannot destroy my pyramid, right? So this is really important. And I think this is really going to be powerful. This is the direction that we have to take plant breeding. And I think, you know, as regulators, as people influencing policy, I think for the security, global food security, this is what we should be aiming at. We aren't there yet. You know, the regulatory environment is going to have to adapt to this opportunity. But this is where I'd like to see it go. Regulatory issues then. You know, these are still being resolved. We've already seen a little bit of discussion. I think the stakes are very high. Whether it's GMO or non-GMO, you know, I think we need to treat this as a very precise non-GMO breeding tool as much as possible. But the inevitability is we are going to have some level of regulation, and that's rightly so. But if you think about globally, you have, as Becky was saying, there are some countries that are process-based, right? So there's the EU, Argentina. I'm not sure with Brazil. I thought it was process-based, but you say it was the other one. Oh. We're not, fortunately, in Brazil at the moment. Okay, but on the other side, you've got product-based. And I think this is much more rational. You should be regulating things on what's in the field and what the consumer is actually being delivered, right? Not how you got there. You know, it's a philosophical difference. It can be highly controversial. But if you're looking at the product, it doesn't then matter whether it's detectable or not. You have a product, right? And that's what it should be judged on in food safety and things like that. The USDA says changes that could be achieved by conventional plant breeding will not be regulated. That's where they are at the moment. So they're saying a small genetic change, which you could have got by mutation breeding or some translocations and things like that, that's okay. That won't be regulated because, to be honest, it's indistinguishable from classical plant breeding. There is an issue that if a pathogen was involved in the delivering of the editing reagents, which some strategies do, the USDA says, you know, they may like, it's a little bit unclear whether they're going to be regulating that or not. So what needs to be considered then, small genetic changes versus allelic substitutions, that is changing sequence, but to something that could have been done, like very, very precise backcrossing, but then gene introductions, and particularly if we want to make gene stacks, you're taking a gene into a new genetic position, which it'd be really tough to do that by classical plant breeding. And particularly if we start to take genes from outside the sexual compatibility barrier. You know, there's a lot of potential for improving crops with that, but then I think it will be regulated. And that's fine. But you regulate it on the basis of the product, not on the basis of how you got there. If it's treated non-GMO, as Becky was saying, it really decreases the dollars and the time to release. So if society wants to have a responsive system for increasing global food security, they've got to reduce the barrier to adoption of this technology. So I've summarized a lot of, I think, genomic opportunities here. We probably don't have a lot of time to go through all of these. You have the slides. I think I want to recognize, well, the point I want to make on this slide is that precise genome editing is just part of everything that we have on the table. There are bottlenecks and challenges. Again, I've listed them. The last one at the bottom there is germplasm and information exchange. If I can do that on the final slide. Can I just ask here, how many people are aware of the Convention of Biological Diversity in the Nagoya Protocol? Can I have a show of hands? One? Yeah. because it's a great concern of mine and quite a few people, the impact that this could have on global food security. I think it's shall we say, it's very controversial and it's very impactful. The Convention of Biological Diversity is something that's extremely worthwhile. It has 3 major goals. That is the preservation of biological diversity. As we know, a lot of species are being eradicated and lost. It's extremely important that we preserve biological diversity. It would also, the goal is to increase the use of that biological diversity. And finally, there should be fair and equitable benefit sharing accruing from the use of that biological diversity. And I mean, no one disagrees with these 3 goals. I think they're excellent. The problem comes with the Nagoya Protocol and how they're implemented. The Nagoya Protocol is implemented on a country-by-country basis, and it requires agreements with each source country that owns that biodiversity. And it requires tracking and use of germplasm. And also the reach through, it goes on in perpetuity. It goes on, there's no time limit. The unintended consequences are that reality is it's severely restricting the use of germplasm in conventional plant breeding. I cannot use germplasm from the best germplasm collection in the world because it's in Holland. The US is actually not a signatory to the Nagoya Protocol. But if I use germplasm, I have to track the use. And when I release breeding lines, I have to demand that the breeding company tracks it. And then they have to come back and say how much my germplasm was worth in their germplasm. I mean, it's a mess. And therefore, because it's not being used, there is reduced benefits, right? And funds for germplasm. So this is, the unintended consequence is it's restricting the use of germplasm, and the benefit sharing isn't happening. And also the other problem is some countries see the benefit as being benefit in cash. Many of the more developed countries are arguing the benefit is actually the advanced cultivars that you are receiving. I mean, Brazil is one of the proponents of this, but they get tremendous benefit from the advanced corn breeding lines, right? Corn is not endemic to Brazil, nor is soybean. So there is tremendous benefit to these countries through the efforts of the breeding companies, and trying to quantify that is proving extremely difficult. Now, with the new technologies of high throughput sequencing and editing to some extent, and DNA synthesis, another unintended consequence is, so the Nagoya Protocol is driven on the fact that you've got to get seed. And when you get seed, you have to sign some paperwork. Now with knowledge, you don't necessarily have to do that. You can say, okay, if you don't want me to use your seed, I won't. I'll synthesize that piece of DNA. So what the response from the NGOs is to say that we should include DNA sequence information within the Nagoya Protocol. And this is extremely controversial, because how do you track a knowledge DNA sequence? And potentially you'd never be able to do an experiment. I mean, already it's impacting basic research. And I just want to make you aware, this is an extremely controversial and very difficult issue. And I feel personally it's one of the biggest threats to global food security if we cannot use the natural genetic resources, particularly using genome editing. So I think that's my last slide. Yeah, so happy to take questions.
Great. Really appreciate the information. I know we'll have questions. Frank, do you want to start?
It's a great presentation. So my question is, so many advances in agriculture have come through the hybridization of varieties and that revolution.
Yeah.
Are we going to go back to open pollinated type of, and we won't have to use the hybridization, crossbreeding, because it creates so many problems with seed production in third world countries and seed supplies?
I can answer that in 2 directions. I think it's going to make hybrid production more efficient, right? So there are different sort of methods of making hybrids. But interestingly enough, there's something called apomixis, which is hybrids are made, but when you self-hybrids, everything falls apart. You get a lot of genetic segregation. But with apomixis, you actually make a hybrid, and then it doesn't undergo that meiosis that resorts stuff up. It's stable. So you get clonal propagation of the hybrid through seed.
Okay, so that leads to another problem of plant variety protection laws,
right.
correct? So if companies are going to spend all this money developing varieties and they have no way to protect that, how would that work?
Well, I mean, you have the PVP. I mean, you can protect inbred varieties, and I presume you would use that. I mean, I'm not a patent lawyer, okay? So, but I think the legal reality will adjust to that. I mean, apomixis, there are apomictic crops. I mean, they're clonally propagated crops. There's strawberry, there's grapefruit. I mean, I don't think that's going to be too much of a problem. You're right. I mean, the seed industry is going to have to adapt to that. But, yeah, it's changing.
Can you just say that again?
Sorry.
I don't know if
Sorry.
you can't test for it.
That's a different question. Sorry.
Patent to protect the intellectual property of the company that develops it.
No, you certainly can test. I mean, you can distinguish one cultivar from another cultivar very easily by DNA sequencing. Right. So, I think we're crossing questions here. I want to be quite clear. I'm answering the right question, am I? You just answered my question. Sorry, thanks.
So I have a couple questions. The Nagoya Protocol, does it address the concern that development of a product might be extremely profitable but damaging to the environment?
No, the Nagoya Protocol does not address environmental impact. What it addresses is the fair and equitable benefit sharing back to the source country. And the goal of the Nagoya Protocol is actually to generate funds for preservation of biodiversity back in the source country. So the aim, the desired aim is to decrease environmental degradation and preserve biodiversity.
Okay, thank you. And then two other questions. Why was it named that? And what is a gene drive?
Good question. So I believe, maybe you can help me. Nagoya it was negotiated, Nagoya in 96-ish? Okay. Yeah, so there's been there's something of the parties. What's it called? Convention of the Parties? Help me here. Conference of the Parties, thank you. So it's a big international organization. They meet every 4 or so years. Two. Thank you. Jump in, please. It's it's a UN sort of driven organization. It's incredibly bureaucratic, but it is, trying to get information, and there's all sorts of interest groups here, heavily influenced by NGOs and endogenous peoples, I mean, as it should be rightly. I mean, the challenge with the CBD is that its goals are excellent. It's the unintended consequences of its implementation. And I mean, I particularly would love to see benefit sharing going back to the tropical rainforest and things like that. But the way it's being executed is really decreasing our access to germplasm. And I think that's a tremendous threat. And we need to find a way of achieving the goals of the CBD and the spirit of the CBD. And right now, that's not happening. And so, gene drives. Yeah, I thought about gene drives and putting that in. So, for everybody else, gene drives are one of the outcomes of genome editing, so that you can make edits where you introduce a piece of DNA that is preferentially represented in the progeny, right? So that's the drive bit. If you add on to that piece of DNA something that will be deleterious to the species, right, you can then knock down the species. So, for example, one that's been talked about a lot is mosquitoes. If you make it so that you essentially have a piece of selfish DNA that actually kills off either the male or the female, let's say the female, right? So that no, it'll have to be the male, that's right. So every time there's a mating, there's a preferential fact that most of the males coming out of that mating will die. You can see that the population is going to collapse. Now, it's fairly controversial because what happens if you just take a species out of the environment, right? There's an argument you could make for some species of mosquito that seem to be purely transmitted and live on humans. There's area of dengue and malaria and things like this. There's great potential, but it'd be a vast ecological experiment. There are some people so they don't want to do it, but they are doing some experiments with mosquitoes. Then you could potentially do it with rats or mice, but I mean you take mice out of the environment. I mean that's troublesome, right? But then I've just come back from I was in the Galapagos last April. I mean, rats are devastating the Galapagos, right? You could, and they tried to eradicate using poison rats on some island, but sooner or later they come back. But gene drives could maybe get rid of rats on the Galapagos Islands. But sooner or later someone's going to take a rat from the Galapagos Islands and give it to Paris. And actually, I don't know if you know, but there's actually a group in Paris that is defending the rights of rats to survive in Paris. So, I mean, it's a complicated issue. I mean, you could look at weeds. We could potentially, the yellow star thistle, that's an invasive weed that's, tremendous problems in pastures. We could potentially knock down yellow star thistle. Yet there's a lot of pollinators that rely on yellow star thistle. So, and then there's actually on the other side, in the scientific community, there are those that argue that there would be such a selection pressure that the gene drive would mutate and it wouldn't work. So there's a debate. Actually, some people are saying it's too risky to do. Other people are saying, well, it's not going to work anyway. Does that answer your question on gene drives? We could spend some more time on it, but it's a powerful technology that should be looked at, but with caution.
We're going to need to move on to the next speaker, although
Sure.
Fascinating discussion, and I hope the board's enjoying the...
Sure, I'll be around at lunch, and I'll be very happy to continue the discussion.
Well, thank you, Doctor. Appreciate it. Okay. Our next presentation be John Lindboe, HM Claus. John Lindboe is an experienced senior scientist with a demonstrated history of working in plant biology and vegetable breeding. John is a research and innovation fellow at HM Claus, an international company that leading vegetable seed business dedicated to innovation and sustainable development of the highest quality vegetable seeds. So welcome.
Thank you. Could I get a water? Would it be okay if I help myself? Okay, great. I guess I should have said, may I get a water? Yes, I can, but may I? Thank you for this opportunity. So I work for a vegetable seed company. It's called HM Claus. And I was asked to come here and talk a little bit about the perspective of a vegetable seed company and a little bit about gene editing in the future and maybe some of the things that might happen in the future of this field. So, plant breeding has been evolving like everything. Everything's changing, okay? And things seem to be changing a little faster now maybe than they have in the past, but Plant breeding is basically human-directed evolution of plants. Okay, we've been doing that for thousands and thousands of years, and we've used different tools over those thousands of years as they've appeared. So starting with thousands of years ago, where we just selected the best looking or most attractive to us humans, the most attractive plant we found in the wild, and we save seed from that, and we keep planting it, and we're actually putting things through a bit of a genetic bottleneck and enriching for the genes that gave a phenotype or a combination of traits that we were really interested in. And so most of the time we've been breeding, it's been pretty much blind, random-based, and all selection-based. As time has gone on, we got into the period of making hybrids, and when we started making hybrid seeds, we got an increase in yield and other traits. GMOs came on, in the 90s. Now we have, thanks to colleagues like Richard and others, we have amazing technologies and tools to look at the make genetic tests for plants, and we can use marker-assisted breeding and things like that. But the point is, we view this as a continuum, gene editing as a continuum in the tools that are emerging in plant breeding. And as a vegetable breeding company, we have created a lot of different diversity, amazing diversity in plants and vegetables, and that's all been based on accessing the natural genetic variation and basically making new combinations of genes in plants, and that gives us new combinations of traits. Now, to give you kind of an orientation about the company I work for, H.M. Klaus, in the vegetable seed business in general, it's very, very complex and complicated. Okay, H.M. Klaus has breeding programs in 24 different species of vegetables. We're a global company, but globally, 24 different species of vegetables. Each year, we release about 400 new variety types or new market types in 24 different species. So our current catalog of what we're actively selling in vegetable seeds is well over 1,000, a couple thousand different types of crop types. All right? So when you guys think of vegetables, you maybe think of corn, beans, peas, carrots, onions, okay? Globally, there's more than 70 different species that companies are breeding in, we call the vegetable space, okay? The company I work for, we just do a couple dozen. But there's over 70 that are bred, and there's multiple production methods. There's plants can be grown in a glass house, plastic house, net house, open field. Sometimes they're trained or staked. Sometimes they just let them grow. We're seeing things like vertical farming. That might be a totally new way of farming emerging. So all these different ways of production have different traits that they need in their vegetables, in their types, plant types. There's multiple environments. Some are hot and humid and have different diseases than the drier climates. And then you have the whole issue of, are you growing vegetables be processed, or are they a fresh product that comes to the consumer? So all of those different combinations is why we have to have 400 different kinds of 24 vegetables, okay? So huge complexity. We're a company that makes hybrid vegetable seeds. Most vegetable seed companies are hybrid seed companies. And there are dozens and dozens of vegetable seed companies in California alone. It's not just the big ones that you are most familiar with. Lots of different sizes of vegetable seed companies. And you can think of a hybrid vegetable development as having two phases, okay? The first phase is you make the inbreds, okay? So an inbred is a true breeding line. So the progeny from an inbred will look and act just like the parent. Okay, so it's genetically true breeding. You know what to expect, and you cross two inbreds, and you get a hybrid, okay? Now, how long does it take to make an inbred? Well, typically, we do that through successive iterations of selfing and selection for multiple generations. Typically, it's about 6 generations. So how long does it take? Well, that depends on what vegetable you're breeding. If you're an onion breeder or a carrot breeder, you may get one generation every 2 years, okay? So if you're making a new inbred and you're a carrot breeder, you may start when your kid is in first grade, and by the time he gets to high school, you've made your first, out of high school, you've made your first inbred. And now you can start crossing them and make hybrids. So some crops have very long inbred development cycles. Others, cucumbers, cantaloupes, peppers, tomatoes, by countercycling or putting things, countercycling meaning going south of the equator in the winter, in our winter, or using greenhouses, you can get 2 or 3 generations a year. And so that inbred development time is shorter. Then what you do is you make hybrids, okay? And then after we make hybrids, we spend typically 3 to 5 years, multiple years of trialing, testing, and tasting the varieties we make. And in the company I work for, and I think this is typical for most vegetable seed companies, 99% of the hybrids we make do not make it through the 3 to 5 year testing cycle. They get thrown out because they are not competitive with the commercial varieties. They do not outperform the current varieties. They will not be competitive. We won't make any money. And so we spend an awful lot of time winnowing through in the 3 to 5 years, planting under different environments, exposing the plants to as many different disease pressures as we can to see how they perform, because you need to pick the winner, okay? So that's kind of how the process works. Now, a lot of our genetic diversity in plant breeding comes from accessing the genetic diversity in things like wild species, and that's been mentioned by some of the previous speakers. So let me give you some numbers to kind of orient you. So if you're a tomato breeder, you have your elite domestic tomato line, and you cross it with a wild species like Pinellii or Pimpinellifolium. Those are 2 wild tomato species. Pinellii actually has green fruit, about the size of a blueberry, horrible taste. The leaves are kind of round. They don't look like a tomato at all. But if you cross that, and we have, and other people have, you bring about 7 million genetic changes, single nucleotide changes, into your elite line, okay? And we know this from now that we have these tools that have come out in the last few years where we can do whole genome sequencing. We know that there's millions of changes we're bringing in. And incidentally, we also know that in some of these wild species, there's actually hundreds or thousands of genes the wild versions have that the domestic versions do not have. So through domestication, we've thrown out hundreds, sometimes thousands of genes to turn a wild species into a domestic. So there's been a lot of subtraction of genetics through the process of domestication. Even if you go and cross your domestic tomato with a variety of tomato that's a little bit more familiar, like San Marzano, kind of an Italian landrace, the really elongated sausage-shaped tomato, the breeder is bringing in hundreds of thousands of genetic changes. Okay, so these are all things that breeding companies and breeders do. There's also, and those are all just from crossing plants, okay? There are some lab-based tools that are also used, have been used since about the 1940s. We know that if we grow plants under certain stressful conditions, they will accumulate these small genetic changes at a rate that's hundreds of times higher than they naturally do. And so we do that. Sometimes those environmental conditions are chemical treatments. And in the case of tomato, each tomato plant that goes through this has thousands of random genetic changes, okay? So in all these cases, and this is how breeding has been done for hundreds and hundreds and hundreds of years, we have random, unknown, uncharacterized genetic changes that we are bringing in to our breeding material every time we do this, okay? So if you want to think about off-targets, unintended effects, we've got them on the millions of fold scale. Now, a tool that's emerging in the last few years, gene editing and CRISPR, which has been discussed in mechanistically in some detail earlier, so I won't go into too much detail about it, but it has the potential to introduce one change. And it's one change at one place. It's targeted, it's precise, it's known, it's defined, okay? Incidentally, if you were to just take a tomato and let it self-fertilize, the progeny from that, the next generation, we estimate will have about 6 random genetic changes. It will be genetically different than its parent, okay? So evolution, mutation happens. It's still happening, okay? And we can, we use it in these are the ways that we use it to, in plant breeding. And this gene editing is a different, it's a step change because of the precision and the control we have over how many changes we make and where we make those changes. So on the left, I think it's your left, yeah, that's what we do now, okay? That target that looks like someone shot a shotgun at it, that's what we do now. That's how breeding works. You bring in thousands or millions of genetic changes, and then when you make hybrids, you go through years and years of trialing and testing and tasting, and you throw out 99% of the things that don't perform, that don't have the combination of traits you need, and you come out with a new variety. So what we do now is on the left. What we could potentially do in the future is on the right. You throw a dart and you hit your target. Use gene editing to make one change at one spot. So if you were to incorporate that new tool into the plant breeding variety development pipeline, this is what it would look like. So the inbred development phase is the phase of hybrid vegetable breeding that really gets the benefit, that really benefits from gene editing. So it's especially important, would be especially valuable in things like beetroot or carrots or a lot of your rooted vegetables, which go to have a seed-to-seed generation time of 2 years. In a generation or two, using CRISPR, you could create some genetic variation in a targeted fashion in those plant lines and then get your inbreds much faster. And now you go through your 3 to 5 years of development and testing and before your commercial release. Some other crops, where you have short cycling times of 2-3 years, it might not seem like it's as big a benefit, but it would still have very, very powerful consequences, because when we have to go to these even heirloom or wild species for the trait of interest, we're bringing in millions of other changes, and we have to go through that inbred development process and get rid of those unintended genetic changes that we moved in when we made that cross. So for plant breeders, gene editing methods can be used across a lot of different vegetable crops, and that's one reason why they're of interest. They're also very efficient and precise, so they can reduce the R&D inbred development time, and we can still and will still spend the same amount of time in our testing and trialing and be just as rigorous in that. I already mentioned it has some value for plants with long generation times in particular, and as diseases continue to evolve, gene editing can really help us compete with those types of changes. For example, if we find a wild species, and say a new disease shows up, we find a wild species where we know, have identified the gene that this wild plant has that makes it resistant to this disease, and it may be a single nucleotide change, we can duplicate that change in something like tomato, and in just a generation or two, have done the same thing we would have done by back crossing, except the back crossing would have taken about 8 years, okay? And in a crop like tomato, there's about 950 million DNA bases in its genome. So having the ability to pick one out of 950 million and just change that one is very, very valuable, okay? It's relatively inexpensive, or at least it could be, depending on a lot of things, licensing, regulatory, et cetera. But it's widely available to companies of all sizes. Someone asked the question of, Are people going to be doing this in their garage or something? Yes and no. It's a little harder than the typical garage, okay? It's very accessible to skilled researchers, but you're not really going to do much use with it in your garage, okay? You might just kind of play around the edges, but it is accessible to a wide variety of companies, including very small vegetable breeding companies. So what things are of interest? Disease resistance, as I mentioned. Some of these has been mentioned, downy mildew and some others, so I'll fast forward because I think we're a little bit behind schedule. Consumer traits, brighter colors, better flavors, more sugar in corn, more sugar in your tomatoes, darker red colored tomatoes, higher antioxidants, more carotenoids, reducing the amount of gluten in wheat. These are all things that are being looked at in research groups around the world in terms of crops. And some of those could have consumer interest and consumer benefit if we can integrate them into our variety development programs. The industry, I should have said, a lot of these slides are from the American Seed Trade Association. H.M. Klaus is part of ASTA, the American Seed Trade Association. And so the industry that we belong to, we do have a very, very strong and serious commitment to quality and safety. Again, this just goes over some of the testing and trialing that is typical in a respectable breeding company. And we think that regardless of the techniques that are used, new plant varieties should be and will be, tested based on, for multiple sites in multiple years. There's some places that want to, regulate crops that you're using gene editing based on the process. So I call those people Methodists. They're concerned about the method. They don't care what came out of it, but we just care about the method. Well, we can create, my apologies if I just offended anyone, but anyway, we can recreate and duplicate what is already out there much faster with gene editing, okay? We spend, large breeding companies spend a lot of time and have people that are dedicated to doing nothing but collecting diverse genetics of the different species that they work with. And these people do nothing but collect the germplasm, they maintain the seed, they make sure it's healthy, they regrow it when it starts getting old so they can continuously have viable seed. They characterize it, genetically, what is this plant, genetically or phenotypically, what traits does it have? And now as we're understanding how things work, and we understand the genetic diversity and value these wild species have, we can actually use that diversity much more rapidly with gene editing than with crossing. And as things change fast, it's really an advantage to be able to take those resources and apply them faster. So we think that if you're making a product that you could have made using standard acceptable breeding practices, and I mentioned those at wide crosses, so crossing to wild species, narrow crosses to other domestic or maybe even land races, or even the mutagenesis-based techniques. You can create the same thing that way as you can with gene editing. Why would one method be regulated differently than the other? The safety issues and the challenges are the same. Okay, so our policy position at ASTA is plant varieties developed through the latest breeding methods should not be differentially regulated if they are similar to or indistinguishable from varieties that could have been produced through earlier acceptable breeding methods. So that's all I have. I'll be happy to take any questions.
Well, John, I appreciate the presentation, really informative, and I'm sure once that gets into the growers' hands, I'm sure the prices will come down on seed costs because you'll develop them much quicker.
We could have a long discussion about them.
No, we'll open it up for discussion. We have some time here, so. No questions. Bryce, go ahead. Sorry.
John, thanks for your presentation, and congratulations on having an amazing and diverse portfolio of seeds. Wow, 24 species.
It's
thousands
very
of varieties.
complicated to manage.
John, do you think that it could be a presumptuous or misguided confidence or even arrogance to say that it's exactly the same? Is the seed so well known and understood that the work you're doing creates the exact item? You know, soil is so complex. People believe they understand it, but our knowledge of soil grows and grows and grows. And what we knew, we thought we knew, doubles or triples. And to say it's the exact thing, is that truly what you're saying?
Yes, it's situational, okay? So for example, we know from domesticating some crops, like squash, okay? Wild squash is really bitter. It'll actually make you sick, okay? Which is one of the things we did when we domesticated squash, is we got rid of the genes that made the bitter taste. So if you know that, now you take a wild squash, or a squash that has that bitterness, and you can find wild varieties that have knocked out that gene, and maybe you can even define exactly how they knocked it out. And if I reproduce on a molecular level, yes, exactly the same sequence, then yeah, that's what I mean. That's a situation where it is the same. You cannot distinguish between the two. And if you get into things like, oh, I'm going to move.
We can't distinguish it, but do you think there might be technology that would come in 5 or 10 years where you say, whoa, oh, son of a gun, we couldn't detect that? What we can detect now when we do pesticide residue testing is certainly a lot more detectable than what we couldn't do or could do 10 years ago.
Yeah, no, we're down with the technological advances in DNA sequencing and bioinformatics in the last, say, 5 years especially. No, this is as granular as you can get, is the AGCT sequences. We can't go any smaller than that, because that's where the DNA,
right, but how they were put in or put out, oh, son of a gun, we can see that these were put in through a different way they were put in or out.
No, you can't. There'll be many cases where
and
you
will
cannot
be saying
gene.
that in 5 years and 10 years.
Yeah, I believe so.
Okay, well, I think maybe 10 years ago, some of the comments the folks from the GMO or biotech companies were saying were as precise and exact are now, there's such new science. But thank you, John, for giving me your, and I accept that as a professional and educated, certainly much more than I am.
I have a few questions, and I support what you all are doing, but I'm not sure I totally understand the process. You talked about the precise insertion of a gene or genes into a certain sequence. And when that's done, do you get the desired result for, say, drought tolerance? But when that plant grows, there are also other traits that change as a result of that? Did I understand that right, or did I misunderstand that?
Well, possibly. I'm not sure. I think I might understand what you're asking.
Well, I thought I heard you say that if you insert the drought-tolerant gene into a sequence, and then you grow that plant, you still have trials to go, because there are new effect, or you observe different traits of that new plant.
Yeah, you confirm that it performs. So and there's a spectrum of things that you can do with gene editing. Okay, so it can be as small as adding or removing one base out of 950 million, Or Richard talked about some other things where you could even move a gene from another species and put it in a specific location. Okay, so those two examples are on the two extremes of gene editing, because gene editing is a tool. Okay, now what you do with that tool can be very, very diverse. So when we use mutagenesis to create variation, those are single nucleotide changes that we're making. And we're making thousands of those in each plant randomly, unintended, unknown, and then we screen through those and we find the plant that has a trait we want, and often it has some other mutations that we don't want, and through the inbred development process, we segregate the ones we don't want away from the ones we do. And the only way we know that is we grow them under real-world conditions, and you see how they really perform. And a lot of times you get linkage drag that does not let you keep the trait you want without bringing traits you don't want. So the gene editing can help separate those two. And it can be done, like in some cases, a lot of domestication is removing genes from wild species. And so removing those can be as small as deleting one single nucleotide. If you get into more complex traits, say a disease resistance that involves 5 or 6 or 7 genes, then you're going to have a lot more complicated type of editing or genome engineering you'll need to do. And yeah, every time you do something, you want to make a variety that has the best combination of traits, that the combination is valuable, okay? And that's what that multi-year trialing and testing is, to make sure, did you knock something down a little bit more than you really wanted to? Do you still have a combination that's a winner?
Okay.
I hope I answered.
You answered my question, but I want to ask one more. So then if you have a tomato plant, you've already done the sequencing, so you know what the genetic material is.
Mm-hmm.
When you get the new variety, if you do that again, why can't you modify that to have all the, then there's been mutations.
Right.
So those should show up in the sequencing. Why can't you just eliminate those and leave the one that you inserted?
Because of how because the DNA strands are, they tend to be inherited in blocks, okay? And so if there's a change you want and a change you don't want very, very far away on the DNA, there's a chance that you can get this natural recombination that breaks and dislinks, if you will, unlinks those two. And then you can do that. But there's many, many cases where the changes are so close together on the piece of DNA that the frequency of the one you want and the one you don't want being broken is very, very low. And the other thing I should say is that most mutations, it seems, actually don't have a negative effect. They don't seem to really have any effect, which is why you don't see plants, even though plants every year or generation are accumulating mutations, you don't see them just changing like crazy overnight, because most of those changes don't do anything biologically meaningful. Okay, a lot of them are neutral. Some are negative, some are positive. And negative or positive, this is a human definition of what's positive or negative, okay? A tomato with fruit like this, you might say that's negative if you have to pick tomatoes for a living, okay? If it's this big, that's a positive trait if you're a tomato picker. So this is human directed evolution in terms of, what we're after.
We're running short on time here, but we have a 1 o'clock presentation on Skype, so go ahead.
Okay, just real quick, I have a question more about public perception. How do you think you sell this? CRISPR sounds like there's a lot of opportunity here, but we've seen the pushback with GMOs, and it's somewhat difficult to explain, yet public perception is everything when it comes to marketing agricultural products. And I'm just curious, have you guys thought about how do you educate the public?
Personally, I think what would be just the coolest thing is if there was a tomato that was so good, every one of you wanted to eat it and you would not stop eating. You'd say, I don't care how this was made. This is the best tomato I've ever had, or the best strawberry you've ever eaten. If you can really get something that a consumer tastes and they say, wow, this is better. This is like, you know, a pluot. If you've ever had pluots, like, man, those are amazing. That's what would really, really sell it.
And I agree, but
You
the
know?
problem is the transparency involved. Every consumer today is asking exactly where it came from, what the variety was, where it was grown, what water source and everything. And I'm just curious how we,
No,
take
it's
this
still
further.
going to be a challenge, but if it's, oh, this is a benefit for the grower, it's really hard for the consumer to see, okay, well, I guess the price of corn chips is 2 cents lower than it was last year, so that's a benefit to me. It's hard to make those connections. I think it has to be something that is immediate and obvious to the consumer. And there will still be those issues that you bring up, but that has to be part of the equation, I believe, to really get people hooked into it.
Well, John, thank you very much. I really appreciate you answering our questions and the presentation. So you guys are welcome to join us for lunch. The speakers in the room to the left here as you go out the door. And I'm sure we'll have more questions if you can hang around. Thank you. We need to be online at 1 o'clock here. We have a Skype presentation, so that means everybody please be back in here ready to go at 1. Thank you. Okay, we'll go ahead and reconvene our meeting. At this point, we'd like to welcome Jeff Boss with the Non-GMO Project and Kirsten Wheeler, also with the Non-GMO Project. The Non-GMO Project is a nonprofit organization committed to preserving and building sources of non-GMO products, educating consumers, and providing verified non-GMO choices. Jeff Boss serves as a CFO and Chief Operating Officer, and Kirsten Wheeler is the organization's communications manager. So welcome to our meeting. Great to have you here via Skype. Although
Thank you.
I think it's probably a beautiful day here. Haven't been out for a while.
Yeah, thank you so much. Can you all hear me okay?
We can. Yep. Excellent.
Great. Well, I appreciate that. And Chris and I were talking last night. We decided that I would just hold the mantle for this presentation, and she'll correct me afterwards on anything that I didn't communicate quite right. But thanks for this opportunity to present to the California State Board of Food and Agriculture. Really appreciate it. Thank you for the introduction. So the name's Jeff Boss with the Non-GMO Project. I'm the COO and CFO. Our Executive Director, Megan Westgate, sends her regrets. Unfortunately, was unable to join us. So I'm hoping to cover a couple of topics that we were asked to provide perhaps a bit of a different perspective on, based on what I've seen on your agenda so far today. And so a couple of things. First of all, I'll just start by a little bit about the Non-GMO Project story. And the reason why that's important is to give you kind of a broader perspective on where the need for the Non-GMO Project really kind of came from, and why the project has been so successful so far in the work that we've done. We'll then jump right into a really critical part of the conversation, which is about definitions. And as a standard-bearing organization that running a verification program, definitions are critical to the work that we do. We want to share a little bit about what we're seeing in terms of consumer perspectives and what some of the research out there on consumer perspectives around GMOs and new GMOs. And finally, we're also asked to share a little bit about the regulatory landscape that we're seeing right now. So hope this will probably take maybe 30 minutes, and then there'll be plenty of time for Q&A. I can't see any of you very well. You're very small on my screen, so maybe somebody can help me out when we get to the question time. So as was read out earlier, this is our mission. So to preserve and build sources of non-GMO products, educate consumers, and provide verified non-GMO choices. We do that last thing through our verification program, and many of you are likely familiar with the butterfly logo you see at the top of the screen there. So where did that all come from? So in 2007, the Non-GMO Project was founded by actually a number of U.S. and Canadian retailers, and they started this nonprofit with coming out of real demand from their customers for transparency around GMOs. And this transparency was coming from more awareness around GMO contamination that was happening in the food system of non-GMO sources of food. There were continued long-term health concerns and questions about GMOs, really on the longer term. So there's some questions. Again, this is where the consumers were coming from, and also some level of distrust with big biotech companies. So those factors were kind of coming together where there was this interest and need for greater GMO transparency. And of course, while many other countries around the world, I believe it's about 64 countries around the world that have GMO labeling and regulations, the U.S. and Canada did not at that particular time. Of course, that just changed recently. The other real need was driven from the need for a standard way of defining and measuring non-GMOs within the food industry. And so the need for that industry standard really to create a platform for a rigorous and consistent standard around the definition of GMOs. And so those two factors came together in the formation of the Non-GMO Project. Over the next 3 years, through public comment periods, work with researchers and scientists in the field, the development of the Non-GMO Project standard came together. We're now working actually currently on the public comment period on version 15 of our standard. But once the standard version of the standard was finalized, products started coming to market in 2010. And so those were the first products to really carry the butterfly. Fast forward to 2019 now, we're working with over 4,500 brands that produce products that are verified in our program. And there's over $26 billion in sales of non-GMO project verified products. So why is this all important? Well, when we start to look at what our mission is in terms of providing consumers with the transparency and with choices, you can see this bar chart shows the significant growth in the number of products and therefore choices available to consumers in stores across the U.S. and Canada. And this is reflective of a couple of key bits of timing, I would say, which is the GMO issue was really coming up more and more in the public sphere. There was the timing around this and the move for state-level GMO transparency initiatives and labeling initiatives like Prop 37 in California, which we were heavily engaged in supporting. A lot of that public interest really started having people paying attention to this issue, and that was about the same time that the butterfly was starting to show up on products, and consumers were starting to engage in this conversation around what matters to them. The other contributing factor is that consumers really started to become more and more interested in what is in their food, and at the end of the day, with our mission, what we really believe is that everybody has the right to know what's in their food. And we think that's something that a lot of people can kind of get behind. So as a result of those factors, you can see, here's the growth in the number of products. even today, we were just looking at some research yesterday from 2018 where the LEK Consulting, a management consulting firm that did some consumer research around this, came out saying that non-GMO is the fastest growing consumer or product attribute in 2018. So there's more and more people that are interested in what's in their food and finding ways of engaging with that. And the butterfly, because there are now so many products available, is one way that they're starting to connect with that. So we're also seeing not only, while we initially saw a lot of core natural products and organic products that were getting verified, really that core has expanded out into the more, let's call it more conventional shoppers. And so, as well as a broader number and range of products that are available. So we're seeing, over the past couple of years, a lot of animal-derived products are now coming in to be verified, and we've done a lot of work with that industry to set up really rigorous standards around animal feed and appropriate supply chain visibility to be able to trace back from the animal product, from the yogurt container, back to what the cows ate to produce the milk and all the way back up the supply chain. So we see dairy, meat. We're also seeing lots of growth in plant-based proteins and wholesale ingredients as well. So getting some of many more commodities now being verified as part of a feeding into the non-GMO project verification process and being able to support brands that are looking to make finished products that can get verified quite easily. So this market-based approach is doing two really important things. One is it's helping to change the supply chain, and it's also helping to drive non-GMO innovation in food. And I think it's easy to be excited about this new technology or the new techniques that are being developed. And there's lots of, no doubt, amazing potentials in those new techniques. At the same time, I think it's important to remember that food innovation has been happening for thousands of years, and that there are other ways of driving non-GMO food innovation as well. And so we're starting to see more and more of that because of this market-based approach. So just a quick bit on how do we fit in our system so that you can kind of understand it. So we are a third-party verification program. We set the standard and the rules. We contract with technical administrators who actually work with the client to get the product verified, and then we audit that technical administrator to make sure that they are doing the verification at a sufficient level of rigor to meet the standard. So if you want to get your granola verified, for example, you would work with one of these technical administrators. They would evaluate your product. If you have any high-risk ingredients like corn or soy in your product, there's additional levels of rigor. So we require you to be doing regular and ongoing testing of those products. That's where the testing labs come in. And we also require inspections of your facilities where you process those high-risk ingredients. So this is kind of the ecosystem in which we're operating. And of course, we're then interfacing with the brands that are producing these foods, retailers who are selling these foods, and consumers who are curious and want to know more about GMOs and the non-GMO space. So consumers trust us, and they trust us to be paying attention to these issues and sharing information about the GMO space. And our standard and our program is set up in a way that Consumer Reports, again, and for multiple years now, based on our process for developing our standard and how we execute on that and our third-party integrity, they continue to rank us as a highly meaningful label for consumers wishing to avoid GMOs in the food that they buy. So the consumers trust us, and that's supported by these consumer groups that are evaluating us, and they evaluate other non-GMO standards as well for consumers to know which labels they can trust and how they can connect. So let's jump into definitions. As I mentioned, this is a critical part of the conversation. And, one of the things that we're hearing a lot about is that new techniques like gene editing are significantly different and are not GMOs. And so I want to talk a little bit about why we don't share that perspective. And so what I wanted to start by sharing with you is the Codex Alimentarius definition of biotechnology. And this is really the most authoritative international definition of genetic engineering, which comes from the Codex, and which is, the Codex, if you don't know, is a collection of global food standards that are developed by the United Nations to address safety, quality, and international trade, and it's used by the World Health Organization, the World Trade Organization, and others. This is also the definition that we use when we define what a GMO is, and this is really kind of the crux of it. And the underlined section here, talking about in vitro nucleic acid techniques, including recombinant deoxyribonucleic acid, this is the really important part, because this definition there's nothing in this definition that limits the definition to transgenics. And I'm sure you've talked about transgenics today, but for those of you that weren't paying attention, transgenics is taking genes from one species and moving it into another species. Part of the leap forward in the new genetic engineering techniques is the ability to actually get into the DNA of a species and manipulate it, even at the base pair level, which is in some cases really remarkable. In other cases, though, the process by which you do that falls under this definition, and so because of that. Because of a little bit of that confusion, one of the narratives that's forming is that anything that's not transgenic is not a GMO. And based on this definition and definition that's held up through the UN, that isn't technically accurate. Likely you've talked a lot about CRISPR today. Everyone's really excited about CRISPR. Just wanted to make sure that we're also talking about all of the other range of genetic engineering and gene editing techniques. Here's a list of some of them. We have a research team that continues to stay on top of the new developments and products that are being, and supply chain impacts that are happening with the proliferation of these new technologies. And we really recognize the rapid advancement of these genetic engineering techniques and the ability to create these new GMOs. And we want to make sure to highlight that there's a real distinction between the medical and research applications of these new techniques, which is one thing, and on the other hand, the possibility of an unchecked proliferation of these new techniques in the food system without consent or transparency to consumers. So folks have lots of opinions about GMOs. The question is whether or not consumers should have a right to know the process by which the food is being developed. And so I think making sure that we're talking about this whole suite of new techniques is quite important. And we're not, so in summary on this, we consider, based on this definition, all of these new techniques to be GMOs. We've felt that and known that for a long time, because the definition is the Codex definition, which hasn't changed. I'm not actually sure when it was initially implemented, but it has been quite a long time, and well before these new techniques had really been popularized. So, and we've actually included for almost 2 years now these techniques in our standard. And one of the key challenges that these new techniques pose is that today there aren't commercially available tests. So it's a big change from the old GMOs, which are transgenic, and it's easier to actually detect and test for those. And so there's a whole ecosystem that is out there testing for those old GMOs, but the new GMOs are not yet testable. So we have, for, as I mentioned, for almost 2 years now, required affidavits in our program to ensure that producers are being held accountable to making sure their supply chain for their verified products don't include products of these new techniques. So we require that affidavit. Now, I think it's also important to note that we work closely with the testing community as well, and there is a lot of activity happening right now for the testing community to catch up with these new techniques and come up with new innovations in testing technology to be able to detect the presence of products of these new GMO techniques. And that's going to be very important, especially when you start looking at the new bioengineered disclosure law, which I'll talk about in a second. And I'll just note that we're the only certification that are taking a proactive approach to these new techniques in our standards. Other non-GMO labels and certifications are quite silent on this at this point. So we're not alone in this perspective on these new techniques. In July, the EU, the European Court of Justice, ruled that products of new genetic engineering techniques such as CRISPR, RNAi, and gene drives are to be considered GMOs under European law. What's also interesting is that in the new disclosure law in the U.S., on the one hand, they call out that the new techniques shouldn't be, or the new techniques, the process by which the food product is developed doesn't matter. What matters is what's in the end product. But on the other hand, on their high-risk food list, they include the Arctic apple and the Simplot potato. And the challenge there is that both of those products are actually products of RNAi. And so it really is kind of unclear in terms of what their position is on that, and there's more work to do with the USDA to help educate and get them clear on the position that they want to take. So what are some of how different are the new GMOs? And when you just look at it through the lens of what are some of the core concerns that we've been hearing for years and years about these products? So these are the main ones: novel proteins.
We lost your slides, so they're going to try and get them back up.
Are we all right here? You lost my slides.
We lost your slides, but we still see you.
Okay,
and
great.
hear you.
You can't hear me?
No, we can hear you, but we don't have your slides up. They're working on it right now.
Okay.
But if you want, go right ahead.
I can still going. I could
Sure.
interpretive dance this table for you.
Yep, no problem.
So the point in this slide, I'm not sure if you even got a chance to see it. I'm not sure, and let me know if there's something I can do on my end here. Andy, let me know.
No, we don't have your slides, but we have a much better picture of you.
Oh, well, lucky you.
Just go ahead and keep going. We're good. We don't have your slides, but we'll just listen to you.
You don't have my slides. Okay, so the slide that I'm trying to show, I should just turn my camera towards my screen, is that there are a lot of similar concerns and similar uncertainty about the products of these new GMO techniques as there were with the old GMOs, which include things like off-target effects, as I mentioned. There's unknown health impacts from a long-term consumption standpoint, and this is recognized quite broadly, which is that this is a long-term effects of consuming GMOs is really challenging to actually study. And so there still aren't, to our knowledge, and as of very recently, any long-term studies that have really shown the health implications of GMOs. There are ecological impacts, and which I hope you've spent some time talking about, and there's also this continued risk of contamination of conventional crops, which could be impacting and creating risk to export markets, especially when overseas in Europe, which was taking another firm stance on this. So as far as consumer perspectives on GMOs, I'm really wondering, can I take a second to just try and share my screen again? And because I'm going to be sharing some data now, and let me try doing this.
Maybe.
Are we back?
Not. Yeah, there you go. You're up. All right.
There's that great table I was just dancing for you. Okay, so let's jump into what do consumers think about this. So, and the research I'm going to show you is from the Hartman Group, which has been studying consumer behavior for quite a long time. They produced a report called the Organic and Natural Report. This is the most recent one from 2018. And I'll just highlight a couple of key, I think, perspectives that are helpful to know. And I think this is really important when we start to think about where are consumers. There's a lot of work for all of us, I would say, to educate consumers around this new technology or these new techniques, so that we can be supporting a level of transparency where they're making informed decisions, which is, I think, the goal for all of us. So what they saw was they talked to over 2,200 people across the U.S., across demographics, and these were some of their key findings. 97% said that they're aware of GMOs. So while that awareness is universal, the understanding of what GMOs is actually quite low. And so that's, as I mentioned, where education is really critical. When we actually start to look at, one thing I will mention on this is 46% of those who responded try to avoid GMOs. So 46% of the population actively looking to avoid GMOs. When they started to dig into why do they do this, and an important part about Hartman's approach is they don't just send out surveys to people. They actually follow them through. They send out surveys, then they follow them through grocery stores, look at what they're looking at, ask them questions about why they're picking things up or choosing to buy one thing versus another, and then they go home with them and they look through their cupboards to really align kind of what their aspirational position is versus what their actual true buying habits are. So they do have that extra level of analysis, which is, I think, qualitative analysis, which supports the quantitative. So what are the main reasons to avoid GMOs? So not surprisingly, there is concerns about the possible impact on health and well-being, and that's a general concern we're seeing across the food industry, why we're seeing significant growth in natural and organic and food in general. There's a real need, especially with millennials, to provide a level of transparency in terms of what's in their food. So that links these first two. There's some concerns about health. They want to know exactly what goes into their food. They're concerned about the environment and the potential implications of GMOs on the environment. As I mentioned earlier, some don't want to be supporting companies that use GMOs, don't know enough about them, etc. I think the important thing is that although the primary driver is around health, shoppers are avoiding GMOs for a variety of reasons.
Is that 70% of the 46% that avoid food?
Correct. Yeah.
Okay, so it's about 30% of the total.
Yeah.
Roughly. Okay.
So the key takeaways from this research, and we can share, there's lots more in this report if you're interested, and there are many other reports, but I think this is really poignant in terms of how they dug into this. These are the kind of key consumer perceptions around GMOs. So one, there's generally a discomfort around the idea of natural produce being changed by people into something unnatural. So there's just that discomfort. There's distrust in big business and its motivation for profit in developing these GMOs, and in science and the ability to have all the answers. So I think that's an important thing for us to be paying attention to. And there's also this uncertainty about the consequences of genetic engineering on their bodies and the world. Now, what was really critical to, I think, the conversation you're having today is they also shared a little bit about some of the new genetic gene editing techniques and shared about the difference between, traditional kind of transgenics and these new techniques. And when they tested that, they found that it didn't actually change the perspective on these. So we don't have a lot of data on consumer perceptions around these new techniques because they are quite new. There's much more education to be done to really help understand consumers' perception of it. But this is an initial, and I think a strong data point to suggest that there is just this, there's some general discomfort and distrust of this, of products produced with these technologies. So shifting into the kind of the regulatory space, this distrust is showing up as polls have shown for quite a few years now, that the vast majority of Americans across demographic, across political lines, support mandatory GMO labeling. It was part of the drive for all the state legislations and a big part of the pressure that was put on for there then to be some federal law that would then come and preempt all the state laws. And so that just happened after a long and somewhat painful process. In December, the National Bioengineered Food Disclosure Standard was published. The mandatory compliance date is in January 2022, so there's lots of time to comply. And this law requires mandatory disclosure of genetically engineered ingredients in certain foods. There are a couple things in that statement, which is how do you define genetically engineered ingredients and certain foods. So there are clear exclusions in the program. There's exclusions for highly processed or refined foods and animal products. There's a lot to unpack here. The document itself is 263 pages. We've actually done a webinar recently breaking down the law and the implications. If that's something that you're interested in, we could certainly pass that along. But as far as how it relates to the new GMO techniques, it's really unclear, as I mentioned, whether the GMO ingredients that are derived through, for example, CRISPR and RNA require disclosure. It's really unclear. The law says that there has to be detectable genetic material in the finished product, and that can't be something that could be derived in nature. So all of a sudden it opens it way up, because you could, go in and argue that, oh, well, we're going in and doing gene editing, we're just changing a couple of base pairs, and that's something that could happen in nature. So that's challenging. But we don't even have tests yet to be able to test for these new GMO techniques, so we don't really know how they would be able to test for that. So that's kind of unclear. And as I mentioned, both the apple and the potato on their high-risk lists are created through RNAi. So there certainly are some products that are considered a bioengineered food, which is a new term that they've developed, BE, and which creates a bunch of challenges around the new label that's come out, and the kind of pro-biotech kind of stance that is taken. So we're really unclear in terms of what the implications of that are going to be. From our standpoint, this is the statement that we came out. We also want to really kind of acknowledge that, having worked in this space for over 12 years, this is a really complex issue to deal with. And so while we applaud the effort to move towards mandatory labeling, this law as it stands falls way short of actually being meaningful and really addressing what that 90% of Americans is really looking for, which is true GMO transparency. So in summary, maybe three things to leave you with. One, products of these new genetic engineering techniques are GMOs based on our definition, which relies on the Codex Alimentarius. The drivers for consumers to avoid GMOs don't change with these new techniques. So they don't change from perceptions around the old techniques as of yet. More for us to learn there. And new regulations don't provide meaningful GMO transparency to customers. And so that's what we're really looking for here is, with all of the excitement around this new technology, all the potential medical and research applications, when we start talking about people's food and people's right to know what's in their food, we believe that it is critically important, and we will continue to do our work to help provide a level of transparency that regulators aren't providing today. And we would encourage regulators in a position to be able to provide more meaningful labeling to do so. So with that, thank you so much for this opportunity to present to you, and happy to take some questions.
Well, yep. Thank you, Jeff. We do have questions here. Well, hopefully it'll be smooth. So, Joy, why don't you go ahead?
My name is Joy Sterling, and I'm serving my second term on this board. And my question is, why were the highly processed and refined foods excluded if there wasn't some question about they were somehow connected with GMO? Why would that even come up?
I cannot. That's a great question. It's one of the main complaints about the new law, because a huge amount of the GMOs that are out there are used in highly processed food. So if you are growing GMO corn and you're going to turn that into a corn syrup and you highly process it, or a canola and a canola oil, there's no longer any detectable genetic material in that final product, and therefore doesn't require disclosure. So I can't speak to why, other than
yeah, that's a bad
I can't
thing.
speak to why. I don't get it.
Okay.
Yeah, there's a lot of pushback on that.
And just
Great
a
question.
follow-up on that is that you mentioned distrust of science. That's terribly problematic. Distrust of science?
Yeah. So you're talking about in terms of drivers. So the distrust, I just jumped back to that screen. So the distrust is in science and its ability to have all the answers. So to put a finer point on this, what they found, it's not an overall distrust in science. It's in distrust that science has all of the answers to this, this complexity. So it's a little bit more than that. It's not necessarily a general distrust in science. It's a distrust in the ability for science to have all the answers.
Bryce, go ahead.
Thank you for your presentation. Bryce Lundberg, Lundberg Family Farms.
Hi, Bryce.
My family produces organic rice. I was wondering if organic certification organizations are in agreement with you on the definition and on whether or not these new techniques are GMOs and applicable for organic.
Great. So I think I caught that. So I just want to make sure I got it. So where does organic stand relative to our position on definition in these new techniques?
Yeah, and I was thinking organic. Yeah, so folks like OTA and CCOF, I suppose NOSB would be another one.
Yeah, that's a good question. So I haven't. So, okay, here's what I do know. When we were providing comments on the law, in both cases, we were in contact with the OTA and really looking to align our statements around this. And they look to the similar definition that we do. But I haven't heard them publicly come out and talk specifically about these new techniques. I mean, as we were saying, as I said earlier, we're really, because this is our single issue that we're really focused on, we think it's really important to be out there educating and talking about this right away. I don't know, but I could look into it. I haven't been in touch with them directly on this in terms of whether they have come out with a public opinion on where their stance is with respect to these new GMO techniques.
Will you be doing a bee-free certification? Okay, that's kind of a joke, okay?
A bee, did you say BE free?
Yeah, we're going to do bee-free.
Bee free, yeah.
Thank you, Jeff.
No, we think that, I mean, that's one of the core issues that was raised right away with this new law. I'm not sure if any of you saw the initial logo was a smiling happy face with BE as the eyes, and there was like huge pushback against what was coming across as a fairly biased logo. But in general, you know, so many of the comments were really pushing for using GMO as the term. And actually, this is something that even when you talk to strong proponents of GMOs, they say as well, you know, that it's not helping anybody to be introducing a new terminology at this point. It's just creating additional confusion for consumers and more education to try and catch them up on what is BE and what does it mean. So we're sticking with GMOs.
Hi, Jeff. Nancy Cassidy, retailer. So we heard today from a plant breeder that when a seed is subjected to the CRISPR technology, it is identical, absolutely cannot be determined any difference between that and a seed that has not been altered. Would you comment on that?
I am not a scientist, so I can't comment on that, and it's I think a more nuanced conversation. So if they altered the seed, did they alter the seed? I mean, did they make a change to the seed or not?
They did.
They did make a change to the seed. So in the testing experts that we talk to, they say that it is possible to be able to test for some of these new techniques. It's just not commercially cost effective at this point. And there's a lot more work to be done. So are they the same? I mean, that's one question. One of the questions that maybe this helps. One of the questions that we get a lot is, you know, around animal-derived products. So we consider in our standard that if an animal is eating GMO feed and downstream that's ending up as yogurt, that's not allowed in our program. And the reason for that is because the vast majority of GMOs are grown for animal feed and ethanol as well. And so we're trying to change the supply chain. And my point there is that it's not just the end product that matters, it's the process by which it's developed that's important to people and to consumers as well. So I don't want to step out of my really my area of focus here. But that's the perspective that we take on this. If you're exposing these products to this kind of technology, we're calling it a GMO, and we want to do that so that consumers can have transparency around that and then make their own decisions about whether or not they buy that.
Thank you.
So, Jeff, this is Don Cameron. I farm in California here. But I had a question. Are you finding that that some of your clients may be dealing with a product that really doesn't have a GMO in being grown anywhere? I mean, there's no GMO for that crop, but they want the non-GMO label on their product for, I don't know, I guess just to be clear, but doesn't that kind of throw the ones that aren't labeling into a conundrum as far as they're kind of put in a pile that, well, there's no GMO available for that product, but if they don't have your label, then are they considered, you know, do they have the stigma that they might have a GMO in their product?
Yeah, I see.
Long question.
Yeah. I mean, it's a good question. It's something that we can point you in the direction of a blog that gets into lots of detail on this. Maybe two things quick to say about that. oftentimes one example that comes up is orange juice, right? There are no GMO oranges. Well, if you look at your ingredient panel on your orange juice, there's more in there than just oranges. So in a lot of cases, it's an oversimplified view of what's in the supply chain to produce that product. So that's one angle, which is important. So for example, citric acid, if it's added to the orange juice, citric acid can be and a lot of it is made with GMOs. So there's that first level of digging into the supply chain and what are the actual ingredients. The other part of this is when you think about labels like gluten-free, what we're trying to do is support consumers that are making, very quick decisions in when they're doing their shopping, to know that they can trust the products that they're buying. And we're hearing this more and more from brands, that the butterfly label or things like gluten-free add another level of trust that the brand that's put this together is actually working with third parties to help support the transparency and the supply chain that they're using. And so it helps consumers that are buying non-GMO to when they're looking at new products, on a new shelf, that's one thing for them to be able to cue into, to know, like, okay, this is non-GMO. That's good to know, because as we're expecting to see with these new techniques, we're going to see GMOs popping up much more rapidly than back in the day when we were just dealing with large commodities and big biotech companies. Does that answer your question?
Yeah, that does answer the question, but thank you.
Yeah.
We had another question. No? Yeah, go ahead, Don.
My concern in this whole discussion today has been, it's really about the consumer's choice and what kind of choices they want to make. And I was a little, I wasn't surprised, but on your next to the last slide, you have some language that says you're going to protect the consumer, and which implies that non-GMO food, in my mind, is not safe. And really, to me, your logo is to inform the consumer that it is a non-GMO product. And to say you're looking out for my safety, I guess I'd like to see what the science is, because I think informed choices are important, and if people are not in favor of GMO food, then I can look at the label and make that decision. But for you to say that you're going to protect me, I have a problem with that.
Okay, yeah. Do you want me to just respond to that?
Sure.
Okay, is there a question? Okay. So the question around, does it do enough to protect consumers? Well, so I guess in this case, we're getting down to semantics in terms of what we mean about protect. In this way, what we're talking about protecting consumers from is the proliferation of GMOs in our food supply. And the intent behind mandatory labeling is to provide a level of transparency for all consumers, not just those brands that are voluntarily paying more to get verified through third-party certifications, but instead to try and provide a level of transparency across the food system. And what we're saying here is that if the intent was to provide that information, this law doesn't provide that level of transparency, and therefore is not protecting consumers from the potential proliferation of GMOs in their food. So hopefully that clarifies. We don't. We are just reflecting back the health concerns that we're hearing about when we talk to consumers about this. And there is, as I mentioned, the main health concerns that haven't really, well, there are lots of concerns that haven't been really answered around GMOs. So, I appreciate your perspective that you see this as us protecting consumers from something that may or may not be harmful, and we're really just seeing it from a transparency standpoint, so consumers can make that decision.
Well, Jeff, I really appreciate your presentation and the time spent on our questions here. So thank you very much for joining us this afternoon, and hope to see you again.
Great. Thank you so much. Really appreciate it.
All right, we're going to shift gears a little bit, and we're going to talk about animal agriculture and genomics. We have Dr. James Murray, UC Davis Department of Animal Science. Dr. Murray is the chair of the Department of Animal Science at UC Davis. Dr. Murray's work is focused on two key areas, genetic engineering of mammals and horse genomics. So welcome.
Thank you. Thank you. I'm probably not really going to talk much about genomics because I thought my understanding from having corresponded with Josh after Allison passed the baton to me was to really talk about gene editing, which is something that I've been involved with. And I've been involved with genetic engineering in animals since the inception of the technology in the early 1980s. So I have tremendous experience and mostly have focused on working for genetic engineering and now gene editing in livestock animals for use in agriculture. I've not really worked on the biomedical side much. So I'll try to go through this fairly rapidly. This is kind of a joint slide deck between Allison and I, and hopefully leave more time for discussion. And I will get heavily into the regulation at the end. So when you're looking at biotechnology and how it's used, if you really go to the scientific definition, I suppose, you really would have to consider everything from using yeast to make beer and wine, artificial insemination, embryo transfer, ovum pickup, all of these things constitute the whole realm of biotechnologies, and within that then there's your subset, at least on the animal side, with cloning and then genomic selection, genetic engineering, and genome editing. And I'm not going to go through everything on the bottom of the slide, but you have the slide deck here, but there's a lot of applications in all of these areas. And to this day, the most genetically engineered animal and gene-edited animal would be probably the mouse, and then the zebrafish, which has been entirely done for research purposes. But there are biomedical, there's pharmaceutical products, pest control, and then agriculture food products across a wide range. And I'll get into some of those more specifically in a little bit. One of the things I will mention on the genomic side, the more information we have gained, the more ability we have to carry out selection. I'm sorry I couldn't be here this morning to see if Richard talked about it on the plant side. But this graph is one that Allison put together. She's directly involved in working on genomic selection. And you can see that the average gain has increased significantly when you can imply a panel of selectable markers, genomic markers that are linked to techniques, to traits that you're selecting for, as well as physical measures in the case of the Holsteins, milk quantity, quality, protein, whatever it might be. So the technology has led to a significant increase in our ability to select our animals, which gets into all of our animals and all of our plants are genetically modified. As a geneticist by training, I dislike the term and choose not to use it because I think it's not meaningful. Genetic engineering, I'll touch really briefly on it because gene editing and genetic engineering are two different things, and I'll get into that in a minute. Genetic engineering is the manipulation of the organism's genomes by eliminating, so a knockout, and rearranging specific genes using modern biotechnology, usually recombinant DNA, as was previously defined. And when you're looking at transgenics and GE, you're really looking at moving whole genes or regulatory parts of genes. They don't have to come from a different species. The first transgenic sheep I ever made had all sheep sequences in it. We'd taken control region from one gene and the protein producing region of another gene and put them together to change the expression pattern of a gene, but it was all sheep. But that would still be considered a transgenic. And there are uses on the production side, most cheese is made using recombinant rennin these days. Not all. A lot of plants are genetically engineered, your big agronomic crops. And there have been animals genetic engineered, in fact, before the first plants, but they've never met with regulatory approval other than the Aqua Advantage salmon, which is on the market in Canada, been approved in the US, but it's been banned from being imported or produced in the country, so it's kind of a catch-22 for the salmon. Currently available transgenic livestock, and we recently wrote a review that came out last year, and these are 5 transgenics. You can see when they were made. The first one is the salmon. It was first reported in 1992. It was produced in 1989. It's the only approved transgenic animal for food, for agriculture use in the world. The other 4 have all had substantial research done on them. So in addition to the original paper that presented the creation of the animal, these have all had 5 papers up to our own work, which is the lysozyme transgenic goats that we produced at Davis, where we've published 18 papers on the safety, efficacy, functionality of what we've done. So these are all currently available transgenic livestock, but other than the salmon, none of them have regulatory approval and therefore aren't being used. So getting into gene editing, I don't know how much explanation you got this morning. I do want to give a little bit before I get into some of the ways it's being used in animals. And essentially, gene editing relies on creating a double-stranded break at a targeted location in the genome. And in the past, in order to do a transgenic insert, you had to have a break, but it was random. With the advent of the gene editing techniques in 1995 with zinc fingers, we were able to then to target that. And so, as you can see here on the slide, the cell has many pathways to repair DNA. If you get a double-stranded break in your DNA, the cell has 2 choices. It fixes the break or it dies. That's the only 2 choices the cell has. So you have a lot of redundant mechanisms in which to repair that cell. And the most commonly used one in our somatic cell tissue, non-germline, is NEGJ, which just basically takes any 2 ends of free DNA and sticks it together. And this is how your body seals that break. And that's a natural way of creating mutations. You can get deletions and you can get insertions by doing that. On the other hand, if you provide some sort of donor template, which is shown in blue here, then you can actually have homology-dependent repair. And that can either take the form of simply copying your repair template, which would be a small break, or you can go to the extent of inserting an entire transgene. And so depending upon how you're using the technology, you can end up with a transgenic animal. But I would argue on the non-homologous end joining side, they're not transgenic. So I disagree with the previous speaker. And when you look at that, we've had 3 different systems developed since 1995. The first one is zinc finger, and that is where we worked out the mechanism by which proteins could recognize and specifically bind DNA. And then we could make that protein in the laboratory, we could hook it up to an enzyme domain that would cut DNA, and we could make our own enzyme to cut DNA. So when you use zinc finger as a gene editing tool, you're not injecting any nucleic acid, you're injecting a protein. So it would not fit with the codex definition. That started in 1995. About 10 years later, we got talons. This is another bacterial system in this case, again using a protein alphabet, as it were, to recognize and read DNA. And so by building a specific tail on each strand of DNA, as I've shown here under talon, you can then, in fact, again, specifically target DNA. And again, you have to provide an enzymatic domain, which is the FOC1 nuclease domain. Again, when you use talons to create gene editing, you're only injecting protein. If you're going down the non-homologous end joining route to, say, just cause a mutation rather than an insertion of some kind. So both of those just inject a protein. When you go to CRISPR-Cas system, which we're using and is being probably more heavily used now than the talons, There you're using an RNA guide, which is a nucleic acid, but not DNA, a small amount of RNA, and then you're using a protein again. You can either inject a gene which would produce all of this as RNA and protein, or you can inject the RNA guide and you can, with the protein, they'll complex, and then again, they will specifically target DNA and specifically then cut. Once you've got that double stand break, as I said, it has to be repaired. You can either go the non-homologous end joining or other random repair pathways that are in the cell, or you can go homology-dependent repair, in which case you can either get a conversion when you use a short sequence, or you can actually insert a transgene. So it can go either way. And those are the three current systems. Since CRISPR was first reported about 6 years ago, they've now found, I think at last count that I saw, 5 or 6 CRISPR-like systems from other bacteria. So it may be like restriction enzyme systems where there's going to be multiple different versions of that from multiple different bacterial genre, and that's going to then give you different capabilities. So how might they be used in food animals, which is probably you're most interested in? And these are work that are going on around the world today that Allison and I know of. I'm guessing there's things that we don't know of at present. But in cattle, I'll tell you a little bit more about polled, because we've had the polled animals at Davis, and some of you might have heard of those. And that's essentially trying to introduce the polled allele, which exists in beef cattle, such as Hereford or Angus, into breeds that are horned, Wagyu or Holsteins. Because right now, something like dairy animals, 90% of our Holsteins have horns. They're removed by the veterinarian when they're calves. And that's an animal welfare issue. There's pain, there's cost, there's suffering. But yet having no horns is a good trade, particularly for animals going in and out of a dairy parlor. Keeps them from hurting each other, it keeps them from hurting the workers. So dehorning is necessary, but it can't be done genetically, because if you were to cross your high-producing Holstein with your Angus, you would have a black and white cow that didn't produce milk, or didn't produce good milk, and it would take about 8 to 10 back crosses to a high genetic merit line to bring back up the milk producing capacity. So you'd be looking at somewhere around 20 years, maybe 30 years, in order to introduce it. So in fact, that's why there isn't polled in dairy today. There's a few lines of polled Holsteins, but they're poor milk producers, and therefore the elite breeders don't use them. And if you do that crossing, you're going to destroy your genetic merit. So that's something that's available. Animals are available. In fact, semen from the animals which were at Davis have been transported to Australia. They now have those animals there. They're working with the Australians to introduce it. They're also, the company that owns the cattle are working with Canada as well. Myostatin is a gene that causes muscle growth. So if you knock out myostatin, you'll get a double muscling phenotype. That's been done, last time I checked, in at least 7 countries and in every major livestock meat-producing breed that I know of, including people trying it in fish. Beta-lactoglobulin has been silenced using RNAi and then by CRISPR to eliminate the primary milk allergen that affects young children. Beta-lact, there's multiple allergies that you can have from milk, but beta-lactoglobulin is the main allergen that young kids would be faced with. And that's different to the lactose intolerance and different to being casein, allergic to casein, but it's a major allergen and has no known function in the milk that we're aware of. Interspecies insertion of that SB110 gene is to confer resistance to tuberculosis, so that would be a transgenic approach using CRISPR. Chicken ovalbumin gene has been silenced to remove it from egg. Again, it's an allergen. Insertion of immunoglobulin heavy chain is a germline editing, and it was used to do that, but that's really more of a biomedical use than a farm use. Myostatin gene silencing, again, has been done in the goats. Prion protein has been silenced or deleted. Again, lack of mad cow disease. If you remove the prion, again, the prion doesn't have a known function. You can remove it and the animal is fine. Again, beta-lac has been silenced also in goats, same reasons for cattle. In pigs, I'll show a slide in a minute about CD163 gene silencing. Again, it's been done both in England and in Missouri, and that's to take out the porcine respiratory virus, which is one of the leading viral diseases that affects the pig industry. In the interspecies warthog rella substitution, that's where they're taking an allele. This is an allele conversion approach, taking the allele from the warthog, which is resistant to African swine fever, transferred into domestic pigs. If domestic pigs get African swine fever, your entire herd will be dead in about 7 to 10 days. And it has gotten into Europe from Africa, and it's currently in Russia, and it's currently actually in Western China. And it's causing a lot of devastation because it's essentially 100% lethal. Again, myostatin, like as I said, every meat animal basically has had myostatin. Knockout of the sexual maturity pathways being worked on at Davis, at least. I also know it's being worked on one other place. Again, this is the case, so you'd be able to eliminate castration as a welfare issue. In sheep, the FG-F5 gene silencing increases wool length and yield. So there's a lot of applications, and there's more. We could do other slides worth of applications. These are things that are ongoing at the present. Just to give you a little bit of information about the gene editing and how it would work. This is the African swine fever, and again, essentially, as I mentioned, it's 100% lethal. It's moving into China from the west at the moment. And the way this works is they have identified a single gene mutation, a SNP. In this case, it's a single nucleotide change in the warthog that allows the warthog, it will get the virus, it will get sick, it will then recover, and it will be resistant to the virus for the rest of its life, as opposed to the domestic pig, which gets the virus and dies. And so by collecting eggs from your domestic pig embryo, they're probably collecting one-cell embryos, injecting those embryos with the CRISPR or TALEN-based substrates. You can then get conversion of the domestic pig allele to the allele that's in the warthog. And that was done at the Roslin Institute. And those pigs, they do have a line of pigs now that have the warthog gene, and they will have to be taken to Pirbright, which is their biocontainment level 4 facility, in order to be tested to see if they're resistant to the virus. The porcine respiratory syndrome, which I was talking about, you can see that it's very widespread across the world. Essentially only Australia is free of it. And you can see the dollar cost of that disease. In the pig industry, it is really the largest disease issue in the swine industry. And both of these labs, so Kristen Whitworth and Randy Prather's lab at the University of Missouri and Christine Burkhardt at the Roslin Institute, in both cases independently identified the same cell surface receptor that the virus binds to. And between the two labs, they have knocked it out, and they've also mutated it so that the virus, if it's absent, the virus can't bind and get into the cell. If they mutate it, then the virus can't bind and get into the cell. And basically, those pigs have been tested. The ones from the University of Missouri were sent to Kansas State, where the level 4 containment lab is, and have been tested, and they're resistant. They're not susceptible to the virus. So this is a potential genetic approach to blocking the disease. I've taken some other things out in interest of time that we could talk about. But the point really is that this is a genetic improvement which is permanent and is generational, and so it's a solution to that animal disease, and it helps reduce antibiotic usage and chemical usage, as well as then the cost of caring for the animals, which is a production cost. Myostatin, I couldn't go by without showing a picture. Here you have on the cattle that you're looking at the back ends of are Nelore, so they're a Brazilian cattle. This was done at Texas A&M, and then the pigs were done in China. And again, it just increases the muscle. There are several different known natural myostatin mutations that give varying levels of increasing muscle. So you can tailor it to get what's a production level of muscle that you want without going to the level of something like a Belgian blue, which actually has dystocia issues because of the muscling. The gene editing poll, this is Allison with the blue scarf on, one of her graduate students. These were the two calves that were produced by Recombinetics and then sent to Davis. They should have horns. Genetically, both their parents were horned. The reason they don't have horns is that the Celtic allele from Angus was transferred into these animals using talons at cells in culture, and then they were cloned. So these are actually clones as well. And we raised them at Davis, studied them, bred them. We still have offspring from them at Davis. And semen was collected by the company, Recombinetics, and is being used certainly in Australia. I think it's also in Brazil at this point. So one of the things you have to look at of these techniques, they're cumulative. Breeding, genetic modification of plants and animals occurred as soon as we domesticated them. There are genetic changes between any domestic and its wild relative. And over the millennia of having plants and animals, we've developed breeding goals, we've developed mathematical ways to do selective breeding, which was the progeny testing. We've enhanced that by things like artificial insemination, embryo transfer, genomic selection. And now we have genome editing and genetic engineering, which are a way of adding diversity, adding specific traits in without impacting the broader genome of those animals, which have been selected for the use we have for them. So they're no longer what they were when they were wild. Regulation, I do want to touch on this. I think it's important, and I think it's important in your considerations. Argentina has essentially not looking at process-based. One of the issues that the European Union has is that it's process-based. I can make a transgenic animal in which there's no protein ever produced. All I've done is put in a piece of DNA. We eat DNA in everything we eat. All organic things have DNA or RNA. And so DNA itself is recognized as safe to eat. We digest DNA. We don't take on the traits of the things we eat by absorbing their genes. And so a process-based approach, and that's one of my concerns with the GMO project, is it's concerned with the presence or absence of DNA, which is not what's functional. It's protein, which is functional for the most part. And just having DNA, and that's why my understanding, like in processed oil or processed sugar, sugar is sugar. It doesn't matter whether it comes from a transgenic sugar beet or cane sugar or a non-transgenic sugar beet. It's crystalline sugar is a specific structure. And I think that was the rationale for excluding highly processed. So Argentina is basically saying that if there's a new genetic combination, then it can be regulated as a transgenic. If the method using the transgene is temporary, or there's no permanent insertion of DNA, then it's not subject to regulation. So they're taking the tack that, for instance, the knockout, I'm going to think one of the examples, the knockout of the allele for PRRS in pigs is not transgenic. It's taking out that gene. That would not be regulated under the Argentine rules. Canada has a very similar regulatory approach.
But that's different from the EU, correct? The EU is completely process-based.
The EU has a blanket ban. And the EU, if you read the court ruling, it actually even says mutagenesis techniques are GMO. If you use radiation, and remember there's several thousand plant varieties in agriculture that were produced through radiation, but if it was done before 2001, it's okay. So again, they're having trouble. And remember, Sweden has said CRISPR is not necessarily GMO and won't necessarily be regulated. as per the EU. So there is some difference there. So process-based, I think, makes it difficult versus product-based. The FDA had a guidance for transgenic animals. They put out a draft of that guidance, 187, in 2017 for comment. And essentially what they're now saying, if you intentionally want to alter DNA, it's going to be regulated as a drug. If you get the same alteration in DNA unintentionally, i.e. mutation, it's not going to be regulated. And that's where the FDA is, but they have not released this guidance, even though the comment period ended in June of 2017. So there is an issue there. And again, it's very process-based. It is not product-based. Again, I can genetically engineer an animal such as my goats, where the intent is to put human lysozyme in the milk. But there's no protein product in the meat. The DNA is everywhere, but the protein isn't. And so if you were to eat the meat from those goats, you're not eating any product of the transgene, you're just eating DNA. And I think that's one of the issues with process versus product-based regulation. The USDA statement recently, Secretary Perdue has said that the USDA is not going to regulate plants produced by things like CRISPR as GMO, again, a term I don't like, as genetically engineered. So now the federal agencies are not on the same page in terms of how they're going to regulate, which causes issues because as an animal person, the FDA thinks they have control over what I do, not the U.S. Department of Agriculture. But according to that intentionally mutated thing, this calf would be evaluated as an animal drug because it carries the Celtic allele that's in every polled Angus or Hereford that you've ever seen on the side of the highway that was naturally polled versus one that was dehorned. Whereas the beef cow is not. So it's exactly the same DNA sequence. If it's in my cow because it was intentionally put there, is regulated as a drug. And the offspring are regulated as a drug because the DNA going through meiosis is a drug residue. And in fact, if I do a knockout where I just delete, like in myostatin is just a deletion, the deletion going through meiosis, so the absence of DNA going through meiosis will be regulated as a drug residue, intergenerationally. That's where we're at with regulation in the United States. What's really important is what's at the bottom of this slide. Allison and Elizabeth Maga at Davis in my department, they sequenced these calves to 20 times, so 20 times the genome amount of DNA in the genome to look for off-target mutations. They did not find any. Or what I really should say is you find several million changes, even if you sequence the same animal, which they did 3 times. You will still find several million differences in the genome, and that's to do with the technology. It's also to do with mutation. So, in fact, a CRISPR, particularly a CRISPR-induced insertion or deletion or a CRISPR-based single nucleotide change will, in fact, scientifically not be possible to detect. There will be no way to do that. You can sequence and say there's a difference in the DNA. You will not be able to say that it was caused by a CRISPR or a talon or a zinc finger. And as far as off-target effects, the same problem is there are more. If we were to take any 2 people in the room and sequences, we'd probably have somewhere between 12 and 24 million base pairs difference between any 2 of us. When you're looking at that amount of natural variation that goes through meiosis, off-target effects become very difficult. It's not saying off-target effects aren't there, but they actually occur every meiosis, every generation. No 2 steaks you've ever eaten, no 2 pieces of broccoli are genetically identical. They don't have the same DNA. And that's just science. And of course, I would argue the spotty guy was not a drug. He was a cow. The National Pork Producers Council has put out a position paper on this, and I've highlighted and read the two parts, which I'll read to you. The first part is that the FDA regulatory path will result in a lengthy and expensive approval process and functionally make any gene-edited animal a living animal drug, and every farm raising them as a drug manufacturing facility. And that's the other issue. If they're a drug, and you're going to contain drugs, and you're going to contain the manure from your drugs, and the urine from your drugs, it becomes this issue. And then the last sentence says, the FDA approach is also out of step with the regulatory pathways under development in the rest of the world. Certainly true that they're out of sync with Canada, with Brazil, with Argentina, with Australia, that I know of, and out of sync with Europe, but that would be, well, I wouldn't say it's in a good way. Europe is dysfunctional in that they ban everything, and we're dysfunctional that we don't approve anything on the animal side. Canada has novel-based food regulation. They look at, when you submit an application, is it new? If I was to take Burry's calves to Canada, I could sell them into the meat chain because Canada is going to say, we've been eating the Celtic allele. It is not new, it is not novel, therefore it's not regulated. And in fact, as mentioned here, Recombinetics, which is the company that made Burry and Spotty Guy, they've actually are going to Canada because they can see a pathway forward when in the US they can't see a pathway for their product. So they'll be actually taking their technology offshore, as is Aqua Bounty with the salmon. They're focused on growing their operation offshore. So would gene editing polled Holsteins be subject to additional regulations? In Argentina, no. In Australia, yes, but that's because Australia has a 20 base pair limit on how much DNA you can add. If it's less than 20 base pairs, it's not going to be regulated. If it's more than 20 base pairs, it will. And that's proposed. That's a draft regulation. I don't know if it's been approved yet. Brazil has said no. Canada would be no. The European Union, of course, would be yes, because they've defined everything as GMO. Japan would be no. New Zealand would be yes. And, of course, the United States at present is a yes. And does it really make sense to regulate gene-edited polled dairy calves differently to naturally occurring polled beef calves? As a scientist, I would argue it does not, particularly when the DNA, in the case of putting in the allele from the beef cattle, it's the same DNA.
So, Jim, going back to the U.S. regulation,
Yes.
So isn't the FDA, it's not a final rule, right? There hasn't been a final rule issued, so it's not regulated currently?
That is my understanding.
Okay.
Well, I should say that's the principle under which I'm currently operating. The Guidance 187 is in place for transgenics, but they have not issued a final rule for gene editing. Yeah. That was written in the previous administration, and I think the current administration is less likely to move forward, but on the other hand, being somebody who deals with the FDA on this all the time, we certainly have not gotten movement from them. So there are issues there at the present. This is something that Allison and I recently written, and we've essentially have come out and stated, along with Kevin Wells from Missouri, that the FDA approach is not fit for purpose. It does not regulate food animals in a way that is logical or makes sense, or in fact addresses the issues they purport are there. And as I mentioned before, dietary DNA is generally regarded as safe. It's called GRAS. It's actually an application you can apply for to the FDA, which they, as a matter of policy, simply, not regulation, but as policy, have simply said no transgenic will be given GRAS status, even if it's polled dairy cows or lysozyme in the milk, which are consumed regularly anyway. So, but DNA is safe to eat, again, and it's only DNA in the food product, then there isn't necessarily an issue, or shouldn't be an issue. And one of the other things that we've been talking about for a long time but doesn't get any traction is if we don't use these technologies to increase economic and environmental sustainability of food production, I'm sure you're all aware of the predictions of what the population is going to do. If we truly hit 10 million people in 2050, we will have to produce as much food per year as we've produced since mankind evolved, came out of Africa, per year. From now until back through the millennia, we'll have to produce that much per year with less land, less water, if we have 3 billion more people. So we need to increase the environmental and economic sustainability of agriculture so that it's productive now and it's productive in the future. And if we don't use all the technology at our disposal, then what are we willing to give up? So one of the things that's been absent in the GMO debate, and again, I don't like the word, but I didn't frame it, is what are the benefits? What are the risks and what are the benefits of using the technology in any given circumstance? And not necessarily, and I would argue, we probably don't want cold-tolerant, genetically engineered catfish in the Sacramento River because it's going to destroy the ecosystem. On the other hand, there are other applications where we can have a very positive benefit. And so what are we willing to give up? And here's an example of what we would give up if we had not done artificial insemination. And that was long enough ago. I don't think any of us remember the debate. But post-World War II, when AI was introduced into the dairy industry, you had a lot of the same debate, a lot of the same opposition to using artificial insemination that we have today about using recombinant DNA techniques. And where would we be if we hadn't had AI? And this is production, and if you'll look at the number of cows in the United States in 1940, which was almost 25 million, and it's now down to, way less than the 25 million, and the amount of milk produced. There's been something like a 350% increase in milk with a third the number of cows. That increases the environmental sustainability of dairy. It increases the economic sustainability. We would have given that up if we had chosen not to use AI. Also, we wouldn't have broad-breasted turkeys for Thanksgiving because that industry is entirely based upon artificial insemination. So in summary, there are a range of modern biotechnologies that are used in food animal breeding that do include genomics, genomic-based selection technology, genetic engineering, which is the transgenics, and gene editing. But I would caution that gene editing and genetic engineering are not identical things. Breeders have already used these tools to work on sustainability traits, including disease resistance, animal welfare, drought salt tolerance in plants, product quality. There's a lot of work going on in animals. When the genetic engineering side, there got to be very little work going on in the Western world because the regulatory dysfunction wouldn't allow any products to market. But now with gene editing, there actually are a lot of labs doing work and companies at this point on the grounds that they are predicting the regulatory environment will be better. Ultimately, the biotechnologies complement genetic improvement methods. It is just a technique, and it should be used when appropriate and not used when not appropriate. It's like any other technique. It isn't a silver bullet. It doesn't solve all problems. It also isn't the end of the world and causes all problems. It's just a technique. And lastly is that forgoing safe breeding methods comes with a high potential cost in terms of what do we give up? What benefits do we choose not to use, not to have, if we don't want to use the technology? And I'll stop there for questions.
Dr. Murray, thank you very much for your presentation, pretty enlightening. Do we have any additional questions from the board? Yep, go ahead, Nancy.
I have a simple question.
Yep.
What does polled mean?
Polled means no horns.
Thank you.
So we went from, if you look at an aurochs, which was the foundation animal for cattle, had these massive horns. because they had to deal with wolves and things, down to a longhorn type horn, down to most Angus that you see are naturally polled. They've never had horns, they don't grow horns. Because we protect them, they don't have to protect themselves.
Yeah, go ahead, Bryce.
Hey, Dr. Murray, thank you very much. Very interesting, quite outside my area of expertise, and I'll look forward to investigating these issues further. And by the way, my grandfather on my mother's side was James Murray, and from Straven in just south of Glasgow. Is that the same area?
I'm a Highland Scot.
Okay, very good.
Murray, as you know, is like Smith or Jones. It's
There we go.
everywhere.
What was your position on BGH, and whether all technology related to animal husbandry is acceptable or not acceptable? I mean, certainly on that milk it ended in 2010, and it didn't go into to 2019. You know, that would increase milk production, and yet
It would.
much of the consumer acceptance of that, I think, has or there's been reducing, or I should say, I don't know that. The brands I see have information on them indicating that it wasn't produced with BGH and.
Originally, it was fairly widely used. And I think part of the issue is if you have high-producing dairy cows, you have other issues if you boost them even higher. When I've talked to Marv, our dairy manager on campus, he said he likes to use it for a very brief time to help control energy balance when he's trying to get cows pregnant, but he doesn't see the need to use it as a method to produce production. And so I think the consumer backlash actually is what really probably limited the adoption. But at one point, it was fairly widely adopted if you looked at the amount of sales of the product. But I would say that it's not very widely used today. But I think that's because there's, anybody who's in agriculture knows this, particularly on the animal side, there's a lot of things, and even on the plant side, where you can improve things with management, you can improve things with nutrition, you can improve things, but there's a lot of different ways to get to a result. And with the increasing milk production through selection, I think the need for RBGH, which was not shown to have any health issues for humans.
Was not seen, but the American Cancer Society opposed its approval.
They did.
And it took 3 panels at FDA to get it approved. The first 2 who said no were fired, and then brought in an executive from Monsanto to lead the group to get approval.
Right.
And
I agree with
so that is, I think, part of the concern about these technologies, is that it's confusing to the consumer. And some technology, maybe like the bovine growth hormone, has opposition, has some concern, and yet industry pushes it and gets it approved. And then some may be more benign, and industry pushes it and gets it approved, and consumers don't know how to cut that. And maybe just say, you know what, I just am going to say no, because I can't make the decision when industry or science, or I would say, yes, science is pushing a product for whatever reason, and when they're saying, well, you get the point.
I don't disagree with you. I think things get oversold. There's no question that Monsanto did a very poor job when they first brought out recombinant products, and I think probably because they weren't even thinking about the consumer. They were thinking about the grower, and they weren't thinking about the consumer. I think one of the problems that we have, and I think, again, having been in this realm now for almost 40 years, is that when we blanket distrust science, it leads to the distrust of our government organizations that are there to keep us safe. And so when FDA is there to protect our food supply, as is the USDA. And if we undermine the confidence in them, then we have problems across the board. Is science 100% accurate? No. Science always is revising its opinion based on the newest data. But they do try, and I would argue, I don't know of any instance, even with BGH, where they set out to push a product that wasn't safe, and again, from what I'm aware of on the recombinant side. But it did meet with opposition, and I think partly they did. They didn't call it bovine growth hormone because they didn't want to say they were putting in a hormone. And yet, bovine growth hormone cannot interact with your growth hormone receptor, so it's not like it has an impact. It's not that it was unsafe, but they did. So I do think there are issues, and to the extent that they undermine trust and confidence, it does hurt. And then it hurts adopting things which are useful.
So when does science or regulatory, the regulatory and the science that goes to engage or approve this technology affected by money and politics? And when is it not? I would say often the scientists in the room will say, I'm not affected by money and I'm not affected by politics. It's pure. And is anything pure in this?
I mean, there's no question politics. I think what you're looking at in the opposition movement is largely political and largely about making money. And as is Monsanto in selling GMO. And when I ask my students on campus who owns Monsanto, most of them don't realize it's their parents' retirement. Their parents own Monsanto. And that Monsanto has a responsibility to make money. So there is this push and pull. But I think when the regulatory agency should be trying to operate as pure as possible on facts and on risk-benefit analysis. Again, no scientist is ever going to come out and say there's no risk to anything, right? There's a risk of an earthquake today. There's a risk of getting hit by a bus when you step out onto N Street. There's always a risk, and so one of the issues that you see with the opposition movement, and I took some notes when Josh was speaking from some of the things that I would question, they sometimes don't want there to be any risk. And I would argue that non-GMO verified, which I have no problem with, is a marketing organization. It's marketing. It's selling a service, and it's convinced people to pay them money for them, and God bless them, that's what capitalism is. They can't actually show where there's any scientific risk because there's not been any proven adverse reaction to an animal eating genetically engineered plants, to my knowledge, anywhere in the world that's been documented. Human or animal, either way.
Well, thank you for letting me engage you in that discussion,
Not a problem.
Dr. Murray. I'm
I
sorry
don't
if
disagree
it
with you
creates
necessarily.
an issue there.
All right. Go ahead, Nancy.
I just have a question about that. We hear about the fact that there aren't any studies that have proven, problems, but we never hear about what the studies are. So I just question, who's doing the studies, how many of them have been done, and.
Well, Allison, and I can't give you chapter and verse. Allison did an analysis of a lot of studies done on animals eating genetically engineered, which is published in the Journal of Animal Science. And so that's in meta-analysis because, again, as a scientist, I would not like to see a single study. I think the issue, when the GMO debate started around 1989, 90, somewhere in there, which was about 8 or 10 years after we were doing this, there was a lot of things, well, there had been no feeding trials that this was safe. Well, now there's a lot of feeding trials, but they're short-term. And so now they've gone back to say, well, there's no long-term feeding trials. On the other hand, unless you are eating organic and non-GMO verified and have eaten that all your life, everybody in this room has eaten GMO because about 90% of the non-GMO verified products in the United States would have some GMO in them. And all of your cattle and all of your fish and all of your chickens have been eating GMO corn. And there's no documented human illness ever been attributed to GMO. And part of the problem is, if you define it as the DNA, nobody's ever going to react to DNA. It's if there is a protein product in that that is either allergenic or toxic or something that you would have an adverse reaction. And that just hasn't been demonstrated. Not saying it won't ever occur.
Well, just to follow up, we made the point when we were talking about the plants that there are so many factors that it's hard to single out what may or may not be causal. And so if we could point to asthma rates going up.
You sure can.
You know, so we don't really know if that's a GMO or a GE issue or not. We just assume that there's too many factors to try to figure it out. So I just...
That's true, but I would caution, and one of the things, and again, one of the things, and remember I've been debating people for years on this, if you look at the statistics for, say, autism, and you run a correlation between the introduction of GMO crops and the increase in autism in the mid-1990s, and they just go up, right? Okay, if I do that same analysis with the introduction of this in autism, it goes up. Anything that ABS brakes on your car. So correlations don't make causation. And so when you think there's a problem, you have to break it down and study those factors. And to the extent that people have looked, and there's been a lot of research done, nobody has yet found a causal link between a health problem in an animal or humans and any specific GMO. And again, that's just where the data's at. Might change tomorrow, might be some new papers come out, but so far, it hasn't changed.
Very good. We'd like to thank you for your presentation and sticking with us on the questions here. Thanks again.
Thank you. And you always know where to find us. You're always welcome to come across the causeway and see us. Or invite us over here and we'll come to you.
No, thank you. That's a good way to wrap up. Okay, Josh, board report on
Yes, so real
reporting
quick-
and training. I already know what that is.
It's bad, no. So I put a list in everyone's folder. So basically a reminder that Form 700s are due April 1st. You have a link that has been sent to your email in regards to filling that out. That's the annual notification. I apologize about our system. It's gone kind of crazy, and I know that everyone's getting a lot of notifications saying you're behind, your deadline, et cetera. This is the correct deadline, April 1st. In addition to that, we are required ethics training.
April 1st?
April 1st is not a joke.
That's
It's not
the
a
real
joke.
deal.
And then we also have the ethics requirement as well, and that information is also on this page. A majority of us will be out of compliance within the next couple of months. It needs to be renewed every 2 years. There's only 3 of us that are in the green. Frank.
That's just
Yes.
because he's new.
So the sections that are outlined are the only ones we need to do?
No, you need to do those additional ones. Yes. So there is a requirement that to be a valid certificate, we need to, and be in compliance with AB 1234. Those additional ones you need to select as part of your training when you go through as well. I will send a reminder via email, but just to let you know, we need to take ethics training, and Form 700s are due on the 1st. These are what's officially on record. I can double check.
Yeah, go ahead.
To my knowledge, and I will correct this in an email if I'm wrong. So the Form 700, as a board, I think they are transitioning us to the main state system with FPPC versus with CDFA. And last year we were with CDFA, and I think that login is still saying that we will be in CDFA this year, but I'll verify that.
Will
Because
they-
you'll get, yes.
Will they transfer the information?
Yes, they
to
will
the new
transfer the
system?
information. It all goes to the same place. It's just who the
Thank
report.
God.
Yes, so in theory, if you go to the CDFA link, all your additional requirements, reporting requirements, you might be able to put on there as well and duplicate them. So if you have to report for several agencies, there should be that ability. I'll double check
Right.
on that. But you can copy it, I think, too. So that's the.
Right. A lot.
I'll look.
Thank you, Josh. Appreciate that.
Ethics, make sure you-
Okay, board discussion, any recommendations from this meeting the board would like to put forward? Bryce?
At CDFA, we have a California Organic Product, COPAC, California Organic Product Advisory Committee. And has COPAC taken this issue up as far as this technology relative to organic production in California? I was just wondering if we could check on that and see if we would want to get a recommendation from them or an engagement from them as far as the, or maybe it's just a given under NOSB and that COPAC won't take this up or doesn't need to take it up.
So, that's a good question. So, COPAC, I think it was 3 or 4 years ago, they started a pilot testing on GMO products in organic, very similar to what they do for testing of pesticide residue. And so they did a pilot test about 2 years ago, phased in those results. It was double blind. And then are doing another one this year. They actually just released that, or our staff and our lab just released those results to COPAC at the January meeting just a couple weeks ago at EcoFarm. And I think overall, they gave me the information here just in case we got that question. See, you were right on it, Bryce. To date, we've collected 75 samples. And phase 1 of the pilot had 45 samples, and then the current phase 2 has 30 samples. We've seen a pretty low prevalence in that testing, but that is something that COPAC is being mindful of and thinking about as far as testing more as verification for organic standards and product verification. So they are aware of it, but as far as a policy statement or policy issue, no.
Okay. I was just wondering, we track the federal, right? And
Correct.
And so there really isn't a discussion we need to have at COPAC regarding this technology and its acceptance because we track federal, and whatever the federal says, we do here in California.
Right, exactly.
And I would say the federal still waiting for guidance from USDA because it's still just, there isn't any regulatory on that.
Okay, our last item will be public comment. If you'd like to address the board, we have, step to the microphone, introduce yourself, and we limit the comments to 3 minutes.
Okay.
So please try to keep it there. Thank you.
Sure. Thank you very much. This has been an excellent discussion, and I appreciate all of the really critical questions you all have been asking. My name is Dana Pearls, and I am the senior campaigner of Friends of the Earth US, which is the North American arm of one of the largest environmental networks in the world. And I am pleased to comment to the CDFA about some of the potential threats of gene editing to California's organic agricultural industry. Despite the claims that we may have heard today of precision and potential benefits of genetic engineering techniques like CRISPR, the scientific research has been fairly clear that there is in fact a high uncertainty and number of risks posed by gene editing. There's a report that it looks like is being passed around that should just be the executive summary, which is ultimately a literature review of nearly 180 studies looking at the unintended consequences, mutations from gene-edited plants for agriculture. So please feel free to take a look. In light of the risks, Friends of the Earth recommends a precautionary approach, one which is process-oriented. I know that we've been discussing a little bit of that today, and which assesses and addresses the direct and indirect impacts and risks throughout the whole life cycle of genetic engineering production. These biosafety issues should be fully evaluated before environmental or commercial releases. Meanwhile, we have environmentally and economically sustainable solutions, organic and non-GMO agriculture. Organic sales in the U.S. totaled a new record of $49.4 billion in 2017, which is up 6.4% from the previous year. Counties with high levels of organic agriculture activity lowers a county's poverty rate by as much as 1.35 percentage points. And organic has been said to create an estimated 407,400 jobs in California alone, many of which are more full-time and year-round employment opportunities for farm workers. Additionally, in the face of climate change, studies show that organic agriculture can minimize environmental harms such as loss of biodiversity, soil erosion, and water contamination. I would suggest that gene editing is a risk to organic and non-GMO agriculture. And when I say non-GMO, I am including gene editing in the definition of genetic engineering. So non-GMO would mean non-gene edited as well. We heard from the Non-GMO Project. We also know that this isn't allowed, the gene editing is not allowed in European standards on genetic engineering. And I would argue that we have virtually no regulations that are adequate for these new technologies, such as gene editing in the US. And in the likely case of cross-contamination, this could put California's extensive organic exports to places like the EU at great risk, not to mention our own ecosystems. So I just would offer that we need to protect California's organic agricultural market, our farmers, our processors from the impacts of gene editing. And until there is more science, robust health and environmental assessments, regulations, transparent testing, and clear labeling, there should be a pause on releasing new genetically engineered organisms, such as gene-edited organisms, into the environment or onto the market. And I would like to invite members of the CDFA to learn more about this topic and ask that the use of emerging genetic technologies in agriculture be evaluated for its impacts on organic agriculture, particularly in California, which is a growing and very profitable sector of California's food system. I would also support the suggestion that was just made, which is that there be a coordinated conversation between the CDFA, the California Organic Program, and COPAC to address these concerns, potential assessments, regulations, and developments, regardless of whether organic would allow gene editing. It's something which will affect the organic industry writ large. Thank you.
Thank you, Dana.
Good afternoon, and Don, I thank you for the last meeting, for your deference for some folks that probably used their whole year of public comments, well, maybe a little long, not quite a year of comments. But this week is Black History Month. Yesterday was Rosa Parks Day. It's been kind of an interesting kickoff. You know, I usually don't get sick till the middle of Black History Month because I just run myself ragged. This year, the largest Black Expo is 31 years. This year is going to feature agriculture. After 31 years at Cal Expo, with the farm in the back, we're going to feature agriculture. And so I want to welcome you out there. Josh sent the email to you. I encourage I just sent it. I mean, just like, you know, because I went out, I was begging him. I was like, you're right across the street. Walk across the street. The CEO, founder of Black Expo. He's right across the street. I said, anyway, I encourage you to come, and I'll send some information, and we'll highlight it. Next year, I want to bring a little piece of the World Ag Expo to Cal Expo. Imagine that, and then highlight what California is doing, how we could be Black and farm in California again, and then how we could share this to the world. Thank you.
Excellent. Thank you. Any other comments? Well, we'll have a quiz. We're going to have a quiz after this. I hope everybody took good notes. I feel like I've been back in school today, but really appreciated the program, and and really the perspectives on gene editing and biotechnology. So, Don, you have a... Yes, go right ahead.
I'd like to call a water subcommittee meeting in the next couple of weeks. We have a new governor, and we probably ought to go through some of the policies or positions that we've taken. And so I'll work with Josh. I talked to Don earlier that we probably ought to conference with the secretary before we have the meeting, just so we understand, but there are some real pressing water issues that we need to address. So we'll get that scheduled.
Very good. Thank you, Don. Okay. We are, oh, sorry, Bryce. I'm not going to pull the plug on you.
Well,
Sorry.
thank you. Don, I just wanted to say that I just brought this product from Sierra Nevada. It is called Resilience, and it was produced in response to the Butte County fires. And I would just say, as a resident of Butte County, that there's significant response, and the response still is significant in Butte County, and that if CDFA were able to lend or continues to lend support and Governor Newsom's administration to the effort to respond and then to rebuild the town of Paradise and the town of Magalia and the town of other towns that were significantly impacted by the fire. And I would like to contribute this to the CDFA staff for your enjoyment.
Very good, Bryce. And yeah, the devastation up there was unbelievable, and my heart goes out to all the people that...
Well, not unlike Sonoma, right? And I just appreciate the focus on Sonoma and what Sonoma has done in response, and not unlike what's happened in Shasta and Mendocino and Santa Barbara and Malibu. Yeah, exactly.
No, you're right. Got a lot of devastation throughout the state, so thank you. All right, we're adjourned. Good to see everyone. We'll see you next month.